the autumn of 1992, at Tufts University School of Medicine, Dr. Anna Soto stared at a printout that should have been blank. Her breast cancer cells were multiplying without any added estrogen, dividing as though a hormone had whispered in the culture medium. But the medium was sterile. The water was pure. The only variable left was the vessel holding the experiment, a clear, shatterproof polycarbonate flask, the same plastic that had been molded into baby bottles, incubator hoods, and the unseen lining of nearly every food can in America. The plastic was supposed to be inert. It was not. It was leaching bisphenol A, a molecule built from two phenol rings that fit the body's estrogen receptor, exactly enough to turn on the same genes that govern growth, reproduction, and the architecture of the developing brain. That discovery did not stay in the laboratory. It raised a question that would travel from a mouse colony in Missouri to the Centers for Disease Control, which later found the compound in the urine of 93% of Americans tested, from an industry memo that outlined a campaign of manufactured doubt to the shelves where BPA-free products still carry structural cousins nobody has vetted. The flask was the first signal in a story that is still being written inside every prenatal exam room, every can of soup, every thermal receipt that transfers a hormone mimic through the skin before the ink has cooled. This is the deep archive. We are leaving the upper air now, sealing the hatch behind us. Below the reach of press releases and quick assurances, the evidence waits under pressure. The glass flasks that Soto demanded in her own lab, the Weinberg Group's strategy documents, the FDA's draft assessment that relied on industry data while ignoring everything else, the frozen mouse tissue from Frederick von Sahl's studies that showed a developing prostate changing at doses 1,000 times lower than what was called safe. Listen for the quiet hum of the incubator, the click of a can opener, the hiss of a receipt printer in a darkened retail bay. The archive is already open. We are going deeper now, into the plastic that learned to imitate hormones, and into the system that let it happen. The quiet hum of the incubator was the first sound Dr. Anna Soto registered each morning, a frequency so constant it had become, after 12 years of tissue culture work, indistinguishable from silence. The laboratory at Tufts University School of Medicine occupied a narrow, fluorescent-lit suite on the 4th floor, its windows looking out over the brick and asphalt of downtown Boston. Inside, the air smelled of ethanol and warm plastic, and the metal racks held rows of flat-bottomed culture flasks made of a polymer called polycarbonate. Each flask contained living human cells suspended in a pink nutrient, medium the color of weak tea. Some flasks received a test chemical, a pesticide, a food packaging extract, a compound borrowed from an industrial plant, and some received nothing at all. The ones that received nothing were the controls. The assay itself was called E-Screen, and it had been developed by Soto and her collaborator, Dr. Carlos Sonnenschein, as a tool for detecting estrogenic activity. The logic was borrowed directly from the body's own communication system. Estradiol, the most potent natural estrogen, travels through the bloodstream and binds to receptor proteins inside the nuclei of certain cells, much as a key slides into a lock. The binding is precise and ancient, a molecular handshake that evolved over hundreds of millions of years to govern reproduction, growth, and metabolism. Once bound, the receptor changes shape and grabs onto DNA, switching on a suite of genes that tell the cell to divide. The E-Screen used a well-characterized line of human breast cancer cells that retained this machinery. If a foreign molecule could fit into the estrogen receptor and turn the key, the cells would multiply. The controls, with no added chemical, were supposed to sit in quiescence, a flat baseline against which the hormone mimics would stand out like mountain peaks. But the mountains had appeared in the wrong places. Soto had been staring at the data for weeks, and her frustration had moved through the standard stages of laboratory life. First disbelief, then rechecking of calculations, then suspicion of a technician's error, and finally, the grinding, systematic troubleshooting that consumed months of work. She and Sonnenschein would load a set of plates on Monday, incubate them for 6 days, then count the cells under a microscope on Sunday. In the control groups, the wells that contained cells, medium, and nothing else, the proliferation rates were spiking. In some replicates, the growth was as robust as if they had added a known synthetic estrogen at a concentration designed to mimic a pregnant woman's serum. But they had added nothing. The numbers on the printouts bucked and twisted, making the dose-response curves of their test chemicals unreadable. Their assay, the sensitive instrument they had spent years refining, was deafened by its own background noise. They tore the protocol apart. They tested the water, first the tap water, then the distilled, then the ultra-purified, each source emerging clean. They tested the serum, the growth medium, the antibiotics added to prevent bacterial infection. They replaced every reagent, scrubbed every piece of glassware, autoclaved the pipette tips twice. They changed gloves, changed lab coats, banned anyone from wearing perfume. Still, the phantom estrogen persisted. It was as if the air itself had become hormonal. One afternoon in late 1992, during a long session of elimination, Soto held one of the empty polycarbonate flasks up to the light. The plastic was a hard, amber-tinted material, chosen for its durability and its reputation for biological inertness. It was supposed to be a neutral vessel. But in that moment, a quiet, unsettling thought crossed her mind. What if the neutral vessel was not neutral? What if the plastic, the very container designed to protect the experiment from the outside world, was the source? They devised a simple test. They set up the E-screen in 2 parallel lines, one using the standard polycarbonate flasks, the other using flasks made of borosilicate glass. The glass flasks were heavier, more expensive, and far more fragile. They required different sterilization methods and careful handling. But they were, by all known measures, chemically inert. The cells were seated, the incubator closed, and the 6 days of waiting began. When the counting was complete, the difference was stark. In the polycarbonate flasks, the control cells were proliferating. In the glass flasks, they were not. The phantom had been exorcised, and its exorcism pointed to the plastic. Further analysis identified the migrating molecule, bisphenol A, known in shorthand as BPA. It was the monomeric building block of the polycarbonate polymer, and it was not fully trapped inside the solid plastic. Under the warm, aqueous conditions of the cell culture, tiny quantities of BPA were escaping from the flask walls and dissolving into the medium. And once dissolved, they found their way into the estrogen receptors of the breast cancer cells, mimicking a hormone with a fidelity that was both surprising and, as they would later learn, literally textbook. The paradox settled over the laboratory like the slow fall of dust after an earthquake. The sterile protective vessel had become the contaminant. The material engineered to be a perfect barrier had been, all along, a bridge. A bridge between the industrial chemistry of the 20th century and the intimate signaling pathways of the human body. Soto stood at the sink that evening, washing a set of glass flasks by hand. The weight of the borosilicate glass was solid and cool in her palm, a familiar heft from earlier eras of science. The water ran, and the incubator hummed, and the metal rack stood empty, waiting for a new set of experiments that would be run, from now on, entirely in glass. The plastic flasks, those amber-tinted vessels of convenience, had been relegated to a cardboard box on the floor, destined for a hazardous waste disposal run. But the question that had been born in the flasks could not be disposed of so easily. If a medical-grade plastic, manufactured under strict specifications, could leach an estrogen mimic into a simple saline medium at levels sufficient to drive cancer cell growth, what was happening in the wider world? In the bottles that warmed infant formula? In the epoxy linings of tin cans holding acidic tomatoes? In the sheer, unbreakable shields of incubators that cradled premature newborns? The molecule had a history that stretched back 100 years. Born in a Russian chemist's flask in 1891, tested as a synthetic estrogen in 1930s, London, and then adopted, in the booming post-war years, as the backbone of a polymer industry that valued clarity, strength, and thermal stability above all else. But that history was buried in obscure journals and archived patent files. No one in the plastics industry had asked whether the monomer might speak the body's oldest language. No one in the regulatory agencies had required an endocrine test before granting the substance a blanket designation as safe. The cells, in their glass flasks, had fallen silent. The phantom was gone from the controlled environment of the Tufts laboratory. But out in the world beyond those fourth-floor windows, in the kitchens and nurseries and grocery store aisles, the phantom was only beginning to speak. The discovery did not end the story. It made the story portable. What was this molecule that mimicked estrogen so faithfully, and how long had it been leaching into the world before anyone thought to look? The library copy of the Journal of the Chemical Society had not been disturbed in years. Its spine sighed when Anna Soto opened it to the 1936 volume. She had come looking for a specific table of synthetic estrogens, a paper by a British biochemist named Edward Charles Dodds, whose name she had seen at the end of a chain of citations that ran from her own centrifuged cell cultures straight back into the chemical prehistory of the plastic that was quietly ruining her experiments. The binding smelled of oxidized lignin and the particular stillness of a room where no one opens the old stacks anymore. In the fluorescence of the reading room at Tufts, she found the page, a tidy column of compounds, each one drawn in the skeletal shorthand of the organic chemist, each one tested for its ability to tell a cell to divide, as if an ovarian hormone had whispered in its ear. Bisphenol A was not the most potent entry on that list. It was not the one that would become a drug, a scandal, a congressional hearing, a cluster of cancers. It was simply present, and also ran in a race it had lost before anyone could imagine that the race would be rerun 60 years later, inside a sterile plastic flask in a Massachusetts incubator. The molecule that had forced her cells to multiply without permission had a name, a birth date, and a biography. And the first entry in that biography was not written in a biology laboratory. It was written in a glass reactor in St. Petersburg in 1891 by a bearded Russian chemist who was only interested in whether two cheap liquids could be forced into a single white crystal. Alexander Pavlovich Dyanin, a pupil of the great Butlerov, heated phenol with acetone in the presence of an acid catalyst and let the vapors cool. What settled in the flask was a symmetrical solid, two phenol rings joined by a carbon bridge carrying two methyl groups, the whole structure flat and orderly as a small saddle. He filtered it, measured its melting point, and reported its synthesis to the Berichte der Deutschen Chemischen Gesellschaft under the unromantic name bisphenol. A-dianin had no use for the compound, and no one else would for 45 years. It took its place in Beilstein's Encyclopedia of Organic Chemicals, a multi-volume inventory that served at once as a monument to German chemical method and a cemetery of unanswered possibilities. The white powder had no target in the living body. It was an answer looking for a question. The question arrived not from industry, but from medicine. In London, in the middle of the 1930s, reproductive endocrinology was young and expensive. A gram of natural estradiol, extracted from the ovaries of thousands of sows, could cost more than a physician's annual salary. Edward Charles Dodds, a lean and deliberate physician turned biochemist at the Courtauld Institute, believed that synthetic chemistry could eliminate the slaughterhouse and deliver cheap, stable hormonal drugs. His laboratory screened hundreds of simple phenolic molecules, injecting them into female rats whose ovaries had been removed, and then examining a smear of vaginal cells under a microscope. If the cells changed shape, if they thickened, flattened, became the distinctive scales that announce an estrogenic command, the molecule was active. Bisphenol A was among the compounds they tested. Its two hydroxyl groups, held apart by the rigid carbon bridge at a distance that roughly mimicked the spacing on the natural hormone, allowed it to catch the estrogen receptor, not perfectly, but enough. The rat cells responded. The assay turned positive. But Dodds was not looking for a molecule that merely worked. He was looking for a molecule that shouted. By 1938, his attention had been seized by a synthetic derivative of anise oil, diethylstilbestrol, or DES, a compound whose estrogenic potency dwarfed everything else on the bench. DES could be manufactured cheaply. It survived the digestive tract, and a tin of it could treat 1 million women. In the pharmacological logic of the day, potency was the measure of safety. A drug that worked at a minuscule dose spared the body a high chemical load. Bisphenol A was judged too weak to be a medicine, and Dodds set it aside. He published his findings, including the modest performance of the runner-up, and the note migrated into the medical literature, where it remained as a quiet admission that the molecule could speak the body's language, but not loudly enough to matter. The judgment was entirely rational, and it would, decades later, ring with a dark irony. The very weakness that disqualified it as a drug would make it the perfect invisible passenger in everyday life. The molecule slumbered through the war years and woke in a different world. Polymer science had accelerated beyond anything the 1930s could have imagined, and in the early 1950s, two chemists on opposite sides of the Atlantic began experimenting with the same bifunctional molecule for reasons that had nothing to do with hormones. At the Farbenfabriken Bayer in Leverkusen, a soft-voiced Bavarian named Hermann Schnell reacted bisphenol A with phosgene, a gas whose industrial personality was as vicious as its wartime reputation. Under careful control, the reaction stitched the BPA molecules into long, repeating chains, a polycarbonate emerging from the reactor as a sticky mass that cooled into a substance clearer than glass and tough enough to survive a hammer blow. Schnell recognized what he had made and filed his patent on October 16, 1953. A few weeks later, and 4,000 miles to the west, in Schenectady, New York, a General Electric chemist named Daniel W. Fox was trying to develop a new heat-resistant insulation for electrical wire when he noticed a transparent film forming at the bottom of his polymerization flask. He had combined bisphenol A with diphenyl carbonate, and the accidental film had the same startling clarity and durability that Schnell was seeing in Germany. Fox filed his own patent in November, aware that he was now locked in a race he had not intended to enter. The resulting patent dispute took years to settle, but the outcome was an industrial truce that cleared the way for a material with properties that post-war manufacturing had been aching to find. It was shatterproof yet light, transparent yet heat, stable to nearly 300 degrees Fahrenheit, and it could be molded into shapes that retained their integrity through boiling, freezing, and the daily abuse of a domestic kitchen. The first heat of the polymerization reaction, the sweet, acrid smell of phenols drifting through the pilot plant, the heavy clatter of the molding machine as it slams shut on the first transparent shape. These sensory facts belong to the hidden birth of polycarbonate, but they were quickly buried beneath a vocabulary of purity. The early applications were medical, incubator domes that would not crack if they fell, oxygen hoods that a nurse could see through without distortion, intravenous connectors that were lighter than glass and far harder to break. The plastic was presented as a barrier, biologically inert, chemically stable, a guardian of sterile environments. Then, almost immediately, it spread into the domestic world. Unbreakable baby bottles that could be dropped on a tile floor and boiled for sterilization without turning cloudy. Sippy cups that survived the chaos of the high chair. Food containers that held hot soup without warping. The advertising of the 1950s and 1960s framed a starched nurse handing a newborn through the clear wall of a polycarbonate incubator, or a mother tipping a bottle to an infant's lips, the milk visible in its pristine vessel. The promise was sterility. The subtext was permanence. Permanence, for a polymer, is always a negotiation with the environment. The long chains of polycarbonate slowly shed individual molecules of bisphenol A in the presence of water and heat, a process now recognized as leaching, and the shedding accelerates when the liquid inside is acidic or fatty. By 1957, the same BPA molecule was conscripted into a second industrial career, this time as the raw material for the epoxy resins that began to line the interior of metal food cans. A thin, amber, gold coating was sprayed into millions of cans intended for soup, vegetables, fruit, and infant formula. The epoxy barrier solved a genuine public health problem. It prevented the tin from corroding into the contents. It stopped the metallic taste that ruined canned tomatoes, and it inhibited the growth of Clostridium botulinum, the bacterium whose toxin can kill at vanishingly small doses. It was a real advance, a quiet technology that saved lives and made the food supply safer and more stable. But it also meant that every can of acidic soup, every warm bottle of sterilized formula was in intimate contact with a surface that was gradually, invisibly, releasing a molecule that a different branch of science had already flagged as capable of slipping into an estrogen receptor and delivering a signal. The institutional records of the 1950s describe bisphenol A in terms of mechanical performance, thermal stability, and cost per ton. The patent applications of Schnell and Fox do not mention DODs. The toxicological dossier submitted to regulatory agencies, which granted epoxy can linings a status of safety that opened the entire food supply, measured acute dangers. Did the material kill liver cells in a dish? Did it poison rats at high concentrations? They did not test for the quietest kind of activity, The endocrine mimicry that operates at doses so low they were, in the 1950s, smaller than most laboratory balances could reliably weigh. This was not a conspiracy. It was the architecture of modern specialization. A molecule can travel from a medical journal to an industrial patent without ever passing through a human mind that knows both contexts. The chemist in the pilot plant and the reproductive biologist in the animal house worked in different buildings, read different journals, and were funded by different economies. Between them stood a wall made not of secrecy, but of the sheer volume of knowledge that industrial civilization had already produced. Anna Soto closed the bound journal in the Tufts library and let the pages press together. The table of compounds had not changed, but she now read it, not as a list of data, but as a compressed genealogy of the century she inhabited. The plastic that had taught her cells to divide was born in a Russian academic exercise, tested in a London animal room, dismissed in favor of a more profitable drug, and then adopted by an industry that prized it for properties that had nothing to do with the body. The consequence of that adoption was not a single event, but a slow distribution that nobody had. Charted polycarbonate into hospitals, into kitchens, into landfill leachate, epoxy into every can of beans and pears. Free BPA onto thermal paper receipts that the cashier would hand across the counter with a smile. The molecule that lost the drug race won the industrial one by so wide a margin that it would eventually be detected in the urine of 93% of Americans tested and in the cord blood of newborns before they had taken their first breath of air. The strange part was not that the molecule worked. It was that the two halves of its biography, the endocrine whisper and the polymer backbone, coexisted in separate filing cabinets for more than half a century, until the cells in a plastic flask finally demanded that someone connect them. The first breath of the plastic world arrived, not as a trumpet blast, but as the quiet condensation of a crystal in a flask, the small notation in a scientific journal printed on acid, yellowing paper, the patent filing that did not cite the biology. The can opened by a mother who trusted what she could not see. The industry would soon be producing millions of tons of a substance with a secret endocrine history. And on the day in 1953 when the first heavy molding machine slammed shut around a shape of hot polycarbonate, nobody in the room thought to ask whether the secret would stay buried. The flask was clear. That was the first thing Anna Soto noticed when she held it up to the light in her laboratory at Tufts in the spring of 1992. Clear and hard, with the faint blue-white tint of polycarbonate, the same material that for decades had been molded into baby bottles and incubator hoods, and the golden lacquer inside food cans. The same material that had, she now understood, been leaching a synthetic estrogen into her cell cultures for months, throwing off her experiments with a silent chemical whisper. She set the flask down on the bench and began to trace its history, following the molecule backward from her contaminated cultures to the factory floors and the chemistry patents and the advertisements that had promised a new kind of safety. The story of how that flask came to be in her hand, and in every hospital and home, had begun almost exactly 40 years earlier, in two laboratories on opposite sides of the Atlantic. In 1953, Hermann Schnell, a chemist at Bayer in Germany, and Daniel Fox, a researcher at General Electric in New York, each independently watched a reaction vessel bubble with a new polymer, a chain of bisphenol A and phosgene that cooled into a hard, transparent solid. They called it polycarbonate. It was, in the language of the trade journals, a thermoplastic engineering material. In the language of the men who would sell it, It was a miracle. The miracle had a specific set of virtues. Polycarbonate was shatterproof. You could strike it with a hammer, and instead of splintering into shards, it would absorb the blow and remain whole. It was clear as optical glass, but it weighed half as much. It could be heated to 250 degrees Fahrenheit without softening, plunged into boiling water without clouding, and molded into almost any shape a designer could dream. It did not rust, corrode, or grow brittle with age. In the rhetoric of the mid-century chemical industry, it was the material that had escaped the ancient limitations of metal, wood, and glass. It was the substance of the future, and the future was clean, bright, and perfectly transparent. The hospital was the first to believe. Premature infants, in the years before polycarbonate, were kept alive in incubators made of metal and heavy glass. The hoods were fragile, the seals imperfect, the weight a burden for nurses lifting and cleaning. Polycarbonate changed the architecture of neonatal care. The new incubator hoods were single, seamless domes, light enough to be lifted with one hand, clear enough that the baby inside could be observed from every angle without disturbing the humidified air. The material could be sterilized with steam, autoclaved again and again, and it would not craze. One glossy 1950s advertisement showed a starched nurse in a white cap reaching through a circular port in a polycarbonate incubator, her gloved hands cradling a newborn who seemed to float in a bubble of light. The caption read, "The clear way to protect new life." The promise was not only hygienic, it was visual. Clarity became a synonym for safety. If you could see through the container, the thinking went, there could be nothing hidden inside. From the neonatal unit, the material moved outward. By the 1960s, polycarbonate was being extruded into 4 connectors, oxygenator housings, surgical instrument trays, and the clear plastic tubing that carried blood and saline and medicine into the veins of patients. It replaced rubber and steel and glass in a dozen quiet, life-sustaining applications. Every hospital room became a landscape of clear, hard plastic. The material's appearance of purity was so complete that it stopped being noticed at all. It became the default background of medical care, as invisible as the air. Then it entered the home. The same shatterproof clarity that protected premature infants was marketed to parents in baby bottles. A polycarbonate baby bottle, first introduced in the late 1950s, could be dropped on a tile floor and it would bounce, not break. It could be boiled, sterilized, scrubbed, and it would not cloud. The ounce markings were molded into the side, permanent and precise. Mothers could see the milk level, check for sediment, and know that the container itself was not contributing any taste or odor. The bottle was a perfect servant, hygienic, transparent, indestructible. By 1970, polycarbonate baby bottles were sold in every drugstore in America. They were the premium choice, the modern choice, the choice that said a parent had left behind the heavy, breakable glass of a less scientific era. Food storage containers followed. Polycarbonate found its way into refrigerators and lunchboxes and kitchen cupboards. It was molded into pitchers, tumblers, mixing bowls, and the compartmentalized trays that held frozen dinners. The 1960s kitchen, with its Formica countertops and avocado-colored appliances, embraced plastic not merely as a convenience, but as an aesthetic. Polycarbonate containers were clear and jewel-like, their surfaces so smooth they felt almost wet. They had no memory of previous meals, no lingering stain of tomato or grease. They could go from freezer to microwave, though the microwave itself was still a novelty, without cracking. The material promised a frictionless domestic life, a kitchen where nothing broke, nothing stained, and nothing was hidden. Beneath this visible invasion, a quieter application was spreading through the food supply. The inside of almost every metal food can, the cans that held soup, vegetables, fruit, baby formula, and the condensed milk stirred into coffee, was lined with an epoxy resin. That resin was synthesized from bisphenol A. It was a thin, golden lacquer, baked onto the metal at high temperature, designed to create a barrier between the food and the tin or steel. Without the lining, acidic contents like tomatoes would corrode the metal, leaching iron and tin into the food and eventually causing the can to fail. With the lining, the can remained stable for years on a shelf. The coating was a chemical shield, a molecular guard that stood between modern convenience and spoilage. By the early 1960s, it was the industry standard. Billions of cans were lined with BPA-based epoxy every year. The regulatory framework that allowed this proliferation was built on a simple, fateful assumption. When the U.S. Food and Drug Administration revised the food additive rules in 1958, it created a category for substances that were generally recognized as safe. For materials with a long-established use and no reported acute toxicity, manufacturers did not need to generate new safety data. Polycarbonate and epoxy resins, already in widespread use by the time the law was written, slid through this loophole. The safety tests that did exist were the standard toxicology assays of the era. Feed a large dose to a rat, look for tumors, organ damage, or death. The word endocrine did not appear in the protocols. The concept that a chemical could mimic a hormone at concentrations 1,000 times lower than the level that would cause a cancer was not yet a scientific discipline. It was barely a suspicion. The lock and key model of hormone receptors was still being debated in endocrinology journals, far from the committee rooms where food safety was decided. So the molecule that had been synthesized as a synthetic estrogen in 1936 and then abandoned for the more potent DES entered the food supply without anyone asking whether its hormonal past was relevant. The polymer was the star. The monomer, the unbound BPA that leached from it, was considered a negligible impurity, a trace residue with no toxicological identity of its own. The paradox was already being molded into the very shape of the material. Polycarbonate's strength came from its long, repeating chains, but no polymerization reaction is perfectly complete. A fraction of the BPA molecules, typically a few parts per thousand, remained unreacted, trapped inside the rigid plastic matrix. When the material was heated, or exposed to acidic or alkaline conditions, or simply aged under ultraviolet light, those free monomers migrated toward the surface and into whatever liquid the plastic contained. The migration was slow, measured in parts per billion per hour, but it was continuous. Every time a baby bottle was warmed, every time an acidic soup sat in its epoxy-lined can, a tiny dose of BPA moved from the container into the body. The dose was so small that standard toxicology dismissed it as biologically insignificant. But the dose was also constant, and it had been arriving, unknown and unmeasured, for every single person in the industrialized world. By the early 1990s, when Anna Soto stood in her laboratory with the contaminated flask, that silent migration had been underway for nearly 4 decades. The Centers for Disease Control would later find BPA in the urine of 93% of Americans tested. But in 1992, that biomonitoring was still a decade away. What Soto had was a single, precise observation: her breast cancer cells, cultured in polycarbonate flasks as a control for the real experiment, were multiplying without any added estrogen. Something in the plastic was activating their estrogen receptors. When she switched to glass, the proliferation stopped. The flask was not inert. The marketing had called polycarbonate the clear way to protect new life. But the biology was telling a different story. The clarity was literal, not chemical. The material was speaking the body's oldest language, and no one had been listening. The sterile, clean smell of a freshly molded kitchen container, the faintly sweet scent of warm plastic from a bottle sterilizer, these were the olfactory signatures of a faith that had become so widespread it was no longer examined. The industry had built a world out of transparent promises, and the promises had been accepted because they were so easy to see through. But the cells in Soto's lab were not persuaded by clarity. They were reading the molecular message beneath the surface, the whisper that had been traveling through every nursery and kitchen and supermarket aisle for 40 years, waiting for someone to notice that the container was not empty after all. The contamination that Anna Soto traced to a polycarbonate flask in 1993 was a whisper. By 1997, that whisper had become a roar in a mouse colony at the University of Missouri. In the quiet of the animal facility, the only sounds were the steady hum of ventilation and the occasional soft rustle of a mother mouse rearranging her litter. Frederick vom Saal placed a mouse pup on the dissecting scope's platform, its body no larger than a thumbnail, and began counting the glandular buds that would form the prostate. Under the scope, the tissue was stained, a faint periwinkle, the buds appearing as tiny rosettes. He had done this count hundreds of times before, but today the numbers were off. The pups whose mothers had been exposed to bisphenol A at a dose so low it was measured in parts per billion, the equivalent of a few drops in an Olympic swimming pool, were developing prostates nearly 40% larger than those of the unexposed controls. The results refused to follow the orderly line that toxicology had been drawing for over 4 centuries. For as long as anyone had studied poison, the rule was simple and quantified. The 16th century physician Paracelsus gave the principle its axiom: all things are poison, and nothing is without poison. The dose alone makes a thing not a poison. Toxicology built its regulatory architecture on that foundation. Test a chemical at increasing concentrations from 1 milligram to 100, graph the response, and you get a line angled upward. The more you add, the bigger the damage. This monotonic dose-response model, the assumption that the line never bends back on itself, was the central engine of chemical safety. It set the allowable limits for additives in food, residues in drinking water, compounds leaching from plastic into formula. The machinery ran on the certainty that a smaller dose was always a weaker dose, that danger could be made negligible by dilution. But in the 1990s, a handful of laboratories began to report a different shape. At the lowest doses, far below the threshold where classical toxicology began its measurements, certain chemicals provoked a response that disappeared at mid-range and sometimes returned, inverted, at high extremes. The line was no longer a line. It was a U, or an inverted U, or a shape that resembled a signature signed with a skipping pen. The endocrinologists called this a non-monotonic dose response. The toxicologists called it noise. The regulatory agencies called it an anomaly, too inconvenient to rewrite the rulebook over. In his laboratory at Missouri, vom Saal pinned the plotted data on a corkboard. The prostate weights did not climb with dose. They peaked at the lowest exposure and fell back toward the control averages as the concentration rose. The numbers told him that the old law was not merely bent, but broken. Bisphenol A did not shout. It did not blister skin or burn mucous membranes. At the gram level necessary to kill a rodent, it was practically inert. But at the part per trillion level, a grain of salt dissolved in an Olympic swimming pool, it could slip into the estrogen receptor on a cell's surface and deliver a signal as potent as the body's own hormone. The receptor did not care about quantity in grams, it cared about molecular geometry. A single molecule, precisely shaped, could bind, trigger a cascade, and change which genes a cell expressed. That changed a cell's behavior. That changed a tissue's architecture. That, in a developing animal, changed the blueprint of the body. Vom Saal had been studying the role of fetal hormone exposure in shaping adult reproductive anatomy for over a decade. His lab examined the minute variations in testosterone and estrogen levels within the womb that determined whether a male mouse developed a large or small prostate, or whether a female mouse reached puberty early or late. In 1997, he began adding bisphenol A to the drinking water of pregnant mice at concentrations that produced blood levels equivalent to those already being measured in a large segment of the American public, between 2 and 20 parts per billion. The pups were born, nursed, and grew to adulthood. When Vom Saal's team dissected them at 6 months, the males that had received the lowest dose showed the clearest anomalies: enlarged prostates, reduced sperm production, altered seminal vesicle weight. The highest doses, in some cases, showed effects that were indistinguishable from the controls. The poison was not in the size of the dose, it was in the timing. And the timing was exactly the window when the body was building its architecture. One morning, a technician lifted a pipette and delivered a dose measured in parts per billion to a water bottle. The soft rustle of the mouse colony filled the air, a mother licking a pup whose body chemistry was being silently rewritten. 5 years earlier, and 1,000 miles to the east, Soto had solved the mystery of her contaminated cell cultures by tracing the estrogenic signal to a polycarbonate plastic flask. That discovery, published in 1993, had been the first alarm that a material approved for food contact could interfere with the body's hormonal language. But Soto's flasks had released bisphenol A in amounts that chemists considered trivial, nanograms of the compound leaching into culture medium over weeks. Regulatory toxicologists, steeped in the Paracelsian tradition, looked at those numbers and shrugged. The levels were orders of magnitude below the established no-effect threshold. The contamination should not have mattered, and yet the cells had heard it, dividing as if bathed in estradiol. The flask was the first whisper that the old law was breaking. The safety of food contact materials was, and remains, governed by migration tests. A plastic or resin is bathed in food, simulating solvents at specified temperatures for specified durations. The extract is analyzed, and the total amount of chemical that migrates is used to calculate a human exposure estimate. Then, toxicologists determine the highest dose that caused no observed adverse effect in a chronic animal feeding study. Divide that number by a safety factor of 100 or 1,000 and set a tolerable daily intake. The assumption, as sturdy as the steel lining of food can, is that effects seen at high doses will diminish as the dose shrinks, vanishing somewhere before the line reaches zero. For a chemical like lead or arsenic or benzene, the math works. For a chemical that binds a receptor already tuned to parts per trillion concentrations of natural hormone, the math collapses. The Food and Drug Administration had approved bisphenol A for food can linings in the 1960s based on 2 industry-funded studies that fed rats 7 doses ranging from 1,000 to 10,000 parts per million. At those levels, the rats showed reduced body weight and liver enlargement. The agency set a no-effect level and derived a safe human intake of 50 micrograms per kilogram of body weight per day. But no one had ever tested the chemical at the dose that a fetus would encounter, 1,000 times lower, 1,000,000 times lower, because no one believed that a dose that small could do anything at all. The machinery of chemical regulation was built to detect the crash of a hammer. It was blind to a whisper. By the close of the 1990s, Vom Saal had published his mouse data, and other labs had begun to replicate. The pattern, low-dose bisphenol A, altered brain development, accelerated puberty in females, and predisposed animals to obesity and insulin resistance. The doses were consistently within the range of human exposure. The levels found in the blood of pregnant women, in amniotic fluid, in umbilical cord blood. And consistently, the standard high-dose toxicology studies, the ones that the FDA and the European Food Safety Authority used to declare the chemical safe, showed nothing. The safety tests did not fail because they were done poorly. They failed because they were designed to measure the wrong thing. They were tuned to detect the crash of a hammer. They could not register the click of a key turning in a lock. The question that should have been asked in 1998 was not whether bisphenol A was safe at the doses that killed rats, but whether a regulatory system built to recognize the blunt trauma of industrial chemistry could ever be retrained to hear a chemical that spoke the body's own language. The metal canisters of infant formula sat on supermarket shelves, their epoxy liners glistening under the fluorescent light. The thermal receipt printed for a box of cereal carried a coating of free, unbound BPA that would transfer to a cashier's fingertips within seconds. The sealants painted onto children's molars slowly released the compound into saliva. The machinery of safety had approved all of it because the machinery had a blind spot, the width of a hormone receptor, and the machinery had no self-correcting gear for the blind spot it could not see. The receipt slid across the counter, glossy side up, still warm from the thermal printhead. The inkless numbers, black, sharp, had seared themselves into the paper in the fraction of a second. It took the strip to pass through the printer. Anna Soto took it between her thumb and forefinger. The surface felt slick, almost waxen, the kind of surface that leaves no fingerprint but holds a chemical powder that the body will absorb within minutes. She was buying a can of soup, a tomato bisque labeled in reassuring green script, BPA-Free. The irony was not lost on her. The smooth square of thermal paper in her hand carried a far higher load of bisphenol A than the product she had just carefully selected. In her laboratory at Tufts University School of Medicine, she had spent the last 2 decades proving that this molecule could speak the body's most intimate hormonal language. Now the whole world was holding the evidence in their hands and throwing it away without a second thought. At home, she set the can on the kitchen counter. The metal was cool. The seam, a thin line of epoxy resin. The same resin that has lined the inside of tin cans since 1957. That year, manufacturers discovered that a thin film of BPA-derived epoxy could prevent food from corroding the steel and stop botulism spores from invading the contents. The resin was a miracle of industrial convenience, tough, heat-stable, flexible enough to survive the high-pressure sterilization of a canning line without cracking. But it was never chemically inert. Heat and acidity, precisely the conditions inside a can of tomato soup, accelerate the release of unreacted BPA monomers from the polymer matrix. The longer the can sits on a warehouse shelf, the warmer the summer, the more the leaching continues. A study at the Harvard School of Public Health later found that people who ate one serving of canned soup daily for 5 days saw their urinary BPA levels jump by over 1,200% compared to those who ate freshly made soup. The metal can, the everyday pantry staple, was a quiet delivery system for a substance that had never been tested for safety at those levels. Structurally, bisphenol A is 2 phenol rings connected by a carbon atom with 2 methyl groups, a molecule that looks to the estrogen receptor like a distant relative of estradiol. Ingested BPA passes from the gut to the portal vein where the liver intercepts it. Hepatic enzymes attach a sugar molecule, glucuronide, to BPA, making it water-soluble and marking it for rapid urinary excretion. Within 6 hours, most of an oral dose has left the body. But Soto's thumb and forefinger, still holding the receipt, told a different story. The skin has no liver. When free BPA powder transfers from the paper onto the skin, it slips through the stratum corneum, into the dermis, and directly into the microcirculation. No glucuronidation occurs. The molecule enters the bloodstream intact, as bioactive as the endogenous estrogen it mimics. A Swiss research team demonstrated that BPA applied to the skin of volunteers appeared in their blood at concentrations far higher than the same dose taken orally, because it had completely bypassed the body's chemical filter. The hand that reaches for the receipt is, in physiological terms, a direct port into the hormone stream. Thermal paper owes its popularity to a simple chemistry. Unlike conventional ink, it uses a color-forming reaction between a dye precursor and a developer. The developer, in most receipts, pure bisphenol A, is coated onto the paper as a fine, free-flowing powder. When the receipt printer's heated head strikes the paper, the BPA melts and reacts with the dye, forming the dark letters and numbers. The process is fast, silent, and cheap, which is why by the 1990s thermal paper had replaced carbon copy receipts in nearly every point-of-sale terminal on Earth. But the BPA is not bound into a polymer. It sits loosely on the paper's surface. Chemist John Warner and his colleagues at the University of Massachusetts published a detailed study in 2010 showing that holding a receipt for as little as 5 seconds transferred half a microgram of BPA onto dry skin. If the skin was moist from hand lotion or sweat, absorption increased tenfold. Cashiers, retail workers, and restaurant servers, anyone who handles dozens or hundreds of receipts in a shift, carried urinary BPA levels significantly higher than the general population. A separate investigation found that BPA from receipts can transfer to paper currency, contaminating the entire cash register ecosystem. In the fall of 2008, the Centers for Disease Control and Prevention's National Center for Environmental Health published a landmark paper in the journal Environmental Health Perspectives. The lead author, CDC chemist Antonia Califat, had overseen the development of an ultra-sensitive method combining solid phase extraction with isotope dilution high-performance liquid chromatography, tandem mass spectrometry. In plain language, a machine that could sniff out BPA in human urine down to 0.4 of 1 part per billion. Her team applied the method to 2,517 urine specimens collected from a representative cross-section of the U.S. population aged 6 years and older, gathered through the National Health and Nutrition Examination Survey. The results were stark. 93% of Americans tested positive for BPA. The geometric mean concentration was 2.7 parts per billion, but the range extended upwards. Children aged 6 to 11 had the highest levels, roughly 40% above the average. Adolescents were next. The oldest adults, paradoxically, had the lowest. This was the first time a federal agency had measured the internal dose of BPA across the entire country, and the number 93% was not a hypothesis. It was a census of the chemical nation. Anna Soto read the CDC report at her desk in Boston. The pages were light in her hands, but they held the weight of a truth she had suspected since the autumn of 1992, when her e-screen assay first malfunctioned and she traced the contamination to the polycarbonate flask. Now the contamination was not a laboratory accident. It was a feature of modern life. The 93% meant that BPA was present in hospital nurseries, school cafeterias, and prenatal clinics. The placenta does not block it. Unconjugated BPA crosses from the maternal bloodstream into the amniotic sac, where the fetus floats in a dilute solution of the chemical. A 2010 study of cord blood from healthy newborns found BPA in over 80% of samples, at concentrations approximating those in their mother's own circulation. The chemical that had been rejected as a medicine in 1936 because it was too weak, and estrogen was now, by sheer volume, the most widespread synthetic hormone in the human population. By the time the CDC paper appeared, the BPA-Free movement was already roaring. In 2007, a panic over polycarbonate baby bottles had prompted Walmart, Toys R Us, and Nalgene to pull BPA-containing products from shelves. A new label appeared on water bottles, food containers, and pacifiers, a bright green sticker bearing the words BPA-Free. But the consumer who tossed a BPA-Free sippy cup into her shopping cart received her receipt and drove home with her canned goods had been misled by a marketing triumph. The receipt she crumpled in her purse delivered a dose of BPA that could exceed the amount migrating from a polycarbonate bottle. The can of beans, the aluminum soda pop top, the jar of baby food with its lid sealant, all contained epoxy linings that leached BPA into the contents. The chemical was a chameleon, hiding in 1,000 objects that never carried a warning, and the replacements waiting in the wings, bisphenol S, bisphenol F, were structural cousins that would later prove to be equally disruptive, a cycle that had only just begun to turn. The toxicologist's old axiom, "the dose makes the poison," had been built around acute poisons, not endocrine mimics. For BPA, the dose was not a single event, but a constant, low-level exposure woven into the fabric of daily life. The CDC's survey captured a snapshot, but the real story played out over decades. In the womb, the fetal mammary gland begins to branch and differentiate under the influence of maternal hormones. An errant estrogenic signal from BPA, arriving at the wrong moment, can redirect a few cells into a developmental pathway that will not be noticed until puberty or until a mammogram 40 years later. The fetal prostate, the hypothalamic pituitary axis, the neural circuits that govern behavior and metabolism, all are tuned by estrogen at parts per trillion levels, all potentially scrambled by a chemical the body never evolved to expect. Soto had spent years arguing that standard toxicity tests, which feed high doses to adult animals and look for immediate harm, were blind to these slow, quiet effects. The CDC numbers confirmed that the experiment was already underway, moving inside every pregnant woman, every child. She placed the receipt on her desk, next to a stack of papers, her own 1993 study, Vom Sal's mouse data, the Caliphate report. The afternoon light caught the faint sheen of the thermal paper, the invisible residue that would soon be washed away with soap, but not before some of it had entered her bloodstream, and briefly joined the low, steady hum of the hormone mimic inside her. Soto had learned long ago that the sterile container could not be trusted. Now the whole world was the container, and the whole species the culture. The chemical was already inside nearly every one of us. But what did it mean for the body that was still building itself? The answer did not yet exist in the CDC's bar graphs. It was written in the cells of the next generation, and it had not finished being read. The wind off Lake Michigan in late autumn does not roar. It inserts itself. It finds the gap between a window frame and the sash, the thin space under a library door, and it reminds anyone sitting alone with musty reports that the world outside is still arranging its forces. In 1990, Theo Colborn sat in a cramped archive room at the University of Wisconsin, a stack of wildlife pathology surveys open in front of her, and she listened to that wind. She had been reading for hours. Each folder held a report from one of the Great Lakes field stations, a description of a herring gull colony, a table of hatch rates, black and white photographs of embryos that had never made it out of the egg. The photographs were the hardest to look at. Beaks that crossed instead of aligning, limbs that ended in a club of bone. Inside some embryos, the reproductive tract had grown into a pattern that belonged to neither sex completely, a silent smear of tissue that defied the pathologist's vocabulary. Colborn was not grieving, she was cataloging, and she had been at it for years. Trained first as a pharmacist, then as a freshwater ecologist, she had become one of the few researchers willing to spend a decade asking what industrial chemicals were doing to the top predators of the Great Lakes food web. The official name for her project was the Wildlife and Contaminants Program, and it produced the kind of data that government agencies file and forget. Eggshell thickness, thyroid gland weight, carcass contaminant levels measured in parts per million. But patterns have a way of rising out of tables when you read enough of them. The pattern Colborn kept seeing was a distortion of the reproductive system, not a killing blow, but a rearrangement. Male gulls with testes that looked like ovaries. Female gulls with oviducts that had collapsed halfway through development. And behind the anatomical oddities, something more alarming. The sex ratio of hatchlings in some colonies had tilted so far that 2 females were emerging for every male. The chemicals responsible were not unknown. Polychlorinated biphenols, or PCBs, had been leaking from electrical equipment and industrial discharge into the lakes for decades. DDT and its metabolites still drifted in from agricultural soil, even though the pesticide had been restricted in the United States since 1972. Those chemicals had been studied before, and their toxicology was understood in terms of cancer and liver damage. No one had looked at them as agents of hormonal corruption, because the very idea that a chemical could imitate a hormone at concentrations measured in parts per billion was, to the mid-century regulatory mind, outside the framework. The framework said, dose makes the poison. A high dose kills cells. A low dose is safe. The gulls were telling a different story. The dose that mattered was not the one that poisoned an adult. It was the one that crossed the eggshell and whispered to the embryo on day 14 of incubation, when the gonads were still deciding what to become. Then, from the warm inland waters of central Florida came a second signal. In 1980, a chemical storage facility at the Tower Chemical Company on the shore of Lake Apopka spilled a slurry of dicofol and DDT metabolites into the lake. The immediate kill was typical: fish floating belly up, a stench, a cleanup that never really finished. But what happened in the following years was not typical. By the late 1980s, the alligator population in Lake Apopka had collapsed, not from death, but from a failure to reproduce. Biologist Louis Gillette from the University of Florida began collecting eggs and juveniles. What he found was a biological disassembly of the male phenotype. Juvenile male alligators had phalluses so small that a researcher needed a magnifying loupe to distinguish male from female. Their blood estrogen levels matched adult breeding females. Their testes, when sectioned under a microscope, contained poorly organized seminiferous tubules, the structures that normally produce sperm. The animals were not just sick, they had been rewritten. Gillette's photographs made the case more starkly than any graph. A healthy juvenile alligator penis is normally a pale tapered cone. The Lake Apopka specimens looked like nothing that should appear on a male vertebrate. Colborn, when she saw the images, felt the same cold recognition she had felt in the library. The alligators and the gulls were not random environmental anecdotes. They were two recordings of the same song, a song that was being broadcast in a chemical frequency. The body's hormone receptors could not distinguish from the natural signal. The step from observation to inference is the hardest in science. Colborn knew the risks of premature causation, but she also knew that the coincidences were accumulating too fast to ignore. She began calling every researcher she could find who had documented similar anomalies in wildlife: fish in English rivers with intersex gonads, frogs in Minnesota with multiple limbs, seals in the Baltic with uterine occlusions. The list grew to over 50 species. In the margin of her notes, she wrote a tentative phrase: hormonal mimicry. In July 1991, she convened 21 of those scientists at the Wingspread Conference Center in Racine, Wisconsin. The building was a Frank Lloyd Wright design, all horizontal lines and quiet, integrated rooms. Rain fell steadily against the tall windows during the 2 days of meetings. Around the table sat wildlife biologists, endocrinologists, toxicologists, and one cell biologist from Tufts University School of Medicine who had brought a different kind of evidence. Anna Soto had not been looking at wild animals. She had been trying to grow breast cancer cells in culture, and she had discovered that the polycarbonate plastic flasks in her incubator were leaching a molecule called bisphenol A. That molecule, which the chemical industry had been manufacturing by the million tons for decades, had a shape that allowed it to latch onto an estrogen receptor and trigger cell division as effectively as the body's own estradiol. Soto's story, already familiar to the listeners of this archive, was the final piece Colborn's gathering needed. The field and the flask were describing the same mechanism. The scientists worked late. They exchanged data on a chalkboard. By the second evening, they had produced a 2-page document that would later be known as the Wingspread Consensus Statement. It introduced a new term: endocrine disruption. It warned that a large and growing number of man-made chemicals were capable of derailing the body's hormonal signaling, that the fetal period was uniquely sensitive, and that the existing safety protocols were blind to effects that occurred at low, chronic doses. The statement did not name bisphenol. Specifically, Soto's work was still too new, but the chemical was already taking its place in a larger category. The publication of Our Stolen Future in 1996 took the consensus from Wingspread and gave it a narrative. Co-authored by Colborn, Diane Dumanowski, and John Peterson Myers, the book dedicated an entire chapter to the wild animal evidence, from the deformed gulls to the Lake Apopka alligators. It argued that the human population was now bathed in the same chemical soup, and that the rising rates of certain reproductive and developmental disorders might share an environmental origin. The chemical industry responded with a coordinated campaign, disputing the low-dose findings and insisting that the human body's defenses, liver enzymes, metabolic clearance, would neutralize any threat before it reached a developing fetus. But that defense, as Colborn had already pointed out, was based on a model that had never been tested against chemicals that work through receptors rather than through direct toxicity. The liver does not filter a whisper. So the burden of proof began to shift. The field biologists had done something that laboratory toxicologists had not. They had looked without a preset hypothesis. They had simply documented what was in front of them. And what was in front of them was a silent, continent-wide experiment on the sexual development of wild animals. The plastic that Soto had discovered in her flasks and that had already found its way into the bloodstream of 93% of Americans was part of that same experiment. The first witnesses were not in white coats. They were in boots, standing on a guano-stained rock in the middle of Lake Ontario, holding a dead chick that should have been a male. The narrative of safe, until proven dangerous, now faced a challenge it could not absorb. The proof was accumulating in the wild, in the laboratory, and in the blood of children who had not yet been born. The gulls and the alligators were not a warning about the future. They were a mirror. Colborn, after the conference, returned to her office. The files she had compiled would eventually fill 17 four-drawer filing cabinets. She kept them until her death in 2014. The papers smell faintly of the lake, of damp paper and old ink. They are an unofficial archive of a discovery that the official regulatory bodies were slow to accept. But they are also a monument to a specific kind of scientific patience, the willingness to listen to what the animals were saying long before anyone understood the language. The rain had stopped when the wingspread meeting ended. The scientists walked out into a humid Wisconsin evening, and their conversation trailed into the parking lot. The question that lingered was not whether the evidence was sufficient, but whether the system they were appealing to was even designed to receive it. In the fields that surrounded the conference center, the crickets were calling. The lake lay silent under a covering of mist. And somewhere in the industrial waterways, a molecule shaped almost exactly like estradiol was completing its journey from a factory outfall to the bloodstream of a creature that had not yet learned to speak. In the dim light of a prenatal exam room, the ultrasound transducer moves over a curved belly, and the monitor shows a face still half-formed, a profile that shifts with the pulse of a heart no larger than a walnut. The mother watches the image rise and fall, searching for the shape of a hand, the arch of a spine, the flicker of reassurance. The machine translates sound into a ghost-gray portrait of a future that has not yet taken its first breath. But it cannot show what else is circulating in that warm, fluid world. It cannot show the molecules small enough to cross the placenta, clever enough to open the same locks that regulate the construction of a body. That mother is not an isolated figure. She is one of thousands enrolled in a birth cohort, a study designed to measure what the body carries before birth and what the child becomes years later. She has given blood. She has collected her morning urine in a small plastic cup, the very material that may contain the substance she fears. Her file holds a number measured in nanograms per milliliter of bisphenol A, a value that tells no one what will happen to the child now curled inside her. The first warning that this number mattered came not from human clinics, but from animal rooms several states away, in a laboratory at the University of Missouri. Frederick von Saal had spent the early 1990s studying fetal development in mice, asking how natural hormones program the trajectory of a life. His questions were not about industrial chemicals. They were about the exquisite sensitivity of the window when organs form, when the estradiol level on a single day in gestation could shift the size of a prostate by 10% in adulthood. Then reports began circulating that polycarbonate cages, the very housing that kept the animals, were leaching a chemical into the water, a chemical that the body read as estrogen. Von Saal decided to test a dose that had been dismissed as biologically irrelevant, 2.5 parts per billion in the drinking water of pregnant mice. The dose was so small, it was not supposed to cause any effect. Standard toxicology said, the poison is in the amount. The poison vanishes below a threshold. That was the law. The law broke. When the pups grew to adulthood, the males had prostates that were larger, heavier, with a different cellular architecture. The gland's internal geography had been redrawn. The effect was not subtle enough to be noise. It was consistent and statistically clear. Von Saal published the finding in 1997. The dose was thousands of times below the safety limit. The poison had no threshold. The signal was strongest at the quietest whisper. The mouse pup at birth is no larger than a honeybee, translucent pink, blind, utterly dependent. Yet inside that tiny body, the branching of the mammary ducts was already being laid down. The prostate buds were already taking their shape. The neural circuits were already choosing their paths. A single misplaced hormone pulse in that narrow window was enough to shift the adult architecture. BPA slipped into the estrogen receptor and delivered a command the body obeyed. The gland grew more cells than it needed. The duct retained branches that should have been pruned. The structure was set. What did that look like in a female pup? Her mammary gland became denser, more vulnerable to later tumor formation. In other animal studies, BPA exposure in the womb accelerated puberty, rearranged the timing of the ovarian cycle, and altered the brain's sexually dimorphic regions, those areas that depend on a precise rise and fall of hormones in the very first days. Mice exposed to BPA as fetuses were more hyperactive in open-field tests. They explored less, or they explored more erratically. The behavior was not a pathology in the traditional sense. It was a shift in the population's bell curve, the mean moving a few points left or right. The distance from a mouse to a human child is measured in many millions of years of evolution. But the estrogen receptor, that ancient cellular lock, has barely changed across mammalian time. The key fits. So epidemiologists began to search for the same pattern in humans, even though the search itself was a paradox. You cannot dose a pregnant woman. You cannot randomize a fetus to receive a plastic chemical and measure the result. You must work backwards from what the mother already carries to what the child becomes, tracking thousands of families for years, waiting for the faint signal to rise from the noise. One of the first large human studies came from the Health Outcomes and Measures of the Environment Project in Cincinnati. Researchers measured BPA in the urine of pregnant women, then followed their children into early childhood. The results, published in 2008, showed that higher maternal BPA levels were associated with altered behavior in daughters, more hyperactivity, more aggression at age 2. The same group reported in 2011 that BPA exposure before birth was linked to language delays in girls. The standard language milestones arrived late, not by months, but by enough to shift a child into the lower end of the distribution to trigger a concern. to launch an evaluation. In boys, the patterns were sometimes different, sometimes the opposite, a common feature of endocrine disruption, because the same hormone signal can mean different things in a male and female brain. Across the Atlantic, Shanna Swan at the University of Rochester published a study in 2005 showing that men with higher BPA exposure had poorer semen quality, lower concentration, fewer motile sperm, a greater fraction of damaged swimmers. That finding looped back to fetal life, because the sperm count a man carries for most of his adulthood is set by the number of Sertoli cells produced in the testis before birth. Disrupt that process in the womb, and the adult man will never know what he lost. He will only arrive at a fertility clinic with a number that cannot be explained. These are statistical shadows. No mother with a high BPA level can be told with certainty that her daughter's speech delay or her son's later infertility is attributable to the chemical. The individual story is always a tangle of genetics, diet, stress, and 1,000 other exposures. The public health story is different. It is the bell curve shifting, a 1% change in the population's average sperm count, a 2% increase in the probability of ADHD. These numbers, multiplied across millions of children, translate into thousands of extra diagnoses, thousands of extra medication prescriptions, thousands of couples sitting in an infertility specialist's office, wondering what went wrong. And the body burden is not evenly distributed. The fetus takes in BPA from the mother's blood, but the detoxification enzyme that helps adults break down the chemical, glucuronyltransferase, an enzyme that attaches a sugar tag to BPA and escorts it out, is barely active before birth. The placenta can slow some toxins, but BPA crosses it freely and enters the amniotic fluid. The fetus swims in it. The newborn takes in more through breast milk or formula, and if the formula is reconstituted from a polycarbonate bottle, the dose can spike. Pound for pound, the infant's exposure is orders of magnitude above the adult's. A mother stands at a kitchen counter, warming a plastic bottle. The water inside is hot enough to accelerate the leaching of unreacted BPA from the polymer matrix. She tests the temperature on her wrist, a gesture as old as maternal care, and tips the nipple to the baby's mouth. The milk goes down. The comfort arrives. And with it, the chemical that no one invited, that the bottle's manufacturer never intended to deliver. The safety was not tested in a way that would have asked about mammary duct branching or prostate weight. The test was based on a model built for strychnine and lead, not for a molecule that can exploit the delicate timing of human construction. Vom Saul's mice kept telling the same story. The dose that mattered most was not the highest acute exposure, but the lowest, earliest one, the one that arrived before the organs had finished their morning instructions. The skin of the mother mouse, the warmth of her fur, the licking of the pup's wet body after birth, all conveyed a chemical legacy that the pup never chose. And when that pup grew up and mated, its own offspring sometimes carried epigenetic marks, methyl groups pasted onto DNA that altered gene expression, that traced back to the grandmother's exposure. The chemical was gone from the body in less than 6 hours. The mark remained. The children now in playgrounds are the first generation born into the universal presence of polycarbonate. Their voices carry across swings and slides, a sound of ordinary joy that masks an unprecedented quiet. They are the unwitting subjects of an experiment without a protocol, without a control group, without an endpoint. The results will not be published in a single paper. They will emerge slowly over the next 2 decades in the registries of childhood cancers, in the prevalence curves of early puberty, in the fertility statistics of the 2040s. The irony is that by the time the full signal is visible, the exposure that caused it will have been replaced by another chemical, its effects still unknown, its name printed on a BPA-free sticker. Frederick von Saal kept paraffin blocks of mouse prostate and mammary tissue in a cabinet in his laboratory long after the original studies were done, not because he doubted them, but because he knew the questions would one day be asked with sharper tools. The tissue weights, the animal data, the epidemiological shadows, the urine samples collected from pregnant women in Cincinnati and Los Angeles and New York all say the same thing in a language that regulation is still not built to hear. The dose is vanishingly small. The window is terrifyingly brief. The consequence lasts a lifetime. And in that lifetime, the child who was once a glowing shape on an ultrasound becomes a parent. The chemical memory may travel forward, not as a mutation but as a cellular echo, a persistent alteration in the way genes are read. The most vulnerable members of society, the ones who have no choice and no voice, carry the highest relative load of a substance invented for the clarity of a 400 and the toughness of a baby bottle. The load is not visible on any monitor, but it is there, measured in nanograms, archived in tissue, waiting to be read. The memorandum was typed in 11-point Times New Roman on the letterhead of the Weinberg Group, a consulting firm that specialized in the chemical industry's most delicate problems. It arrived in the spring of 2003 in a plain white envelope addressed to the offices of the American Chemistry Council and its member companies. The subject line was the kind of phrase that could headline any corporate agenda, scoping document, bisphenol A. The content, however, was a strategic blueprint, a systematic campaign to shift the scientific consensus on a chemical that was becoming an escalating liability. The chemical was BPA. The primary obstacle, the memo noted, was a small group of academic researchers whose data kept refusing to align with industry safety assessments. At the top of that list, named explicitly, was Frederick von Saal, a biologist at the University of Missouri, whose laboratory had become a persistent source of inconvenient results. The memo's language was clinical, its verbs passive. It detailed a plan to recruit scientific consultants who could carry the industry's interpretation to regulators and the press. It called for the funding of new studies designed to find no effects, and for placing scientists who published contrary evidence under a subtle yet firm pressure. The objective was not to resolve a scientific question. The objective was to generate enough uncertainty to prevent a regulatory response, a model borrowed, almost point for point, from the playbook the tobacco industry had perfected decades earlier. The strange part was not that such a document existed. It was that the strategy it described was already well underway, a quiet friction already slowing the gears of regulatory science before any public hearing was called. Frederick von Saal had been studying the hormonal choreography of fetal development for more than 2 decades. His laboratory at the University of Missouri was a controlled environment of mouse cages, the low hum of climate-controlled rooms, the sterile gleam of stainless steel instruments. In the mid-1990s, he began a series of experiments that would mark him for the industry's attention. He fed pregnant mice BPA at doses measured in parts per billion, levels mirroring what humans absorbed from can linings and plastic containers. The offspring, when they matured, showed a constellation of changes: enlarged prostates, early puberty, altered brain structures, metabolic disruptions. These were not gross birth defects. They were subtler rewritings of the body's expected script, and they emerged at doses thousands of times lower than what the Environmental Protection Agency considered the safe daily limit. The mechanism that produced these effects defied the foundational assumption of classical toxicology. For centuries, the guiding principle had been Paracelsus's maxim: the dose mixed the poison. More of a chemical meant more harm, less meant less. For most poisons, lead, mercury, cyanide, this was true. But hormones do not operate on a linear scale. The endocrine system is tuned to detect signals in vanishingly small concentrations. A molecule that fits an estrogen receptor can deliver a command at a dose so low it barely registers on a mass spectrometer. At slightly higher concentrations, the same molecule can saturate the receptor, block the signal, and produce the opposite effect, or none at all. This non-monotonic pattern is not a violation of biological laws. It is a property of the system. The endocrine system is not a bucket that fills. It is a radio receiver that picks up specific frequencies, and BPA was a frequency the body understood. Imagine a thermostat set to maintain a room at 72 degrees. A hand resting on it, cool, steady, can trick the sensor into thinking the room is warm, shutting off the heat. The hand exerts no great force, but the result is a cold room. BPA, at tiny doses, plays the same game with developing tissues. The machinery of regulation, built to weigh poisons by the pound, could not detect a whisper that carried a command. It was a fundamental mismatch between the chemical and the system charged with assessing it. The industry's countermove was to produce a parallel body of science that appeared to contradict von Saul's findings. Between 1997 and 2008, chemical manufacturers funded a series of large-scale BPA toxicity studies. The designs were subtly but systematically different from the academic experiments. Von Saul's group used pregnant mice because the fetal stage is the moment of maximum hormonal sensitivity. The industry-funded studies use non-pregnant rats, often administering BPA directly to adult animals and checking for immediate effects, a design almost guaranteed to miss the kind of developmental reprogramming von Saul was documenting. The rat strain chosen, the Charles River 400 SD, was known among endocrinologists for its blunted sensitivity to estrogen. It was, in effect, an animal model selected for its ability to tolerate the chemical. The results formed a pattern too stark to be a coincidence of method. A subsequent literature review by a consortium of university scientists would calculate that more than 90% of independent, government-funded studies identified some biological effect of low-dose BPA. None of the industry-funded studies did. The discrepancy was not about fraud. It was about experimental design calibrated not to detect hazard, but to demonstrate safety. The scientists working for industry were not fabricating data. They were constructing a framework in which the data could only say one thing. This asymmetry put regulators in a bind. The official risk assessment process treated studies as neutral reports of equal weight, weighing carefully aimed industry arrows against a scatter of independent observations and calling the clustered negatives proof of no effect. The campaign was not confined to laboratories and journals. Frederick von Saul found himself under public assault. His research was branded activist science by industry trade groups. Stephen Henches, the American Chemistry Council's chief BPA spokesman, gave media interviews dismissing von Saul's work as inconsistent, limited, and not reproducible, despite the growing number of independent laboratories that were, in fact, reproducing the low-dose effects. At scientific meetings, consultants paid by the chemical industry rose during question sessions to question his methodology and integrity. Letters arrived at his university demanding audits of his animal care protocols. Congressional staffers, sympathetic to the industry, requested volumes of his raw data. The effect was cumulative and wearing. It was not a single blow. It was a persistent erosion. Graduate students hesitated to join a laboratory mired in controversy. Review panels for grant funding, perhaps unconsciously, began to perceive his proposals as riskier investments. The machinery of doubt did not need to win a decisive victory. It only had to keep the controversy alive long enough for the product to remain on the shelves undisturbed. The institutional peak of this campaign arrived in August 2008. The United States Food and Drug Administration issued a draft assessment of BPA's safety. The document, titled Draft Assessment of Bisphenol A for Use in Food Contact Applications, concluded that BPA posed no significant health risk at current exposure levels. The conclusion was not based on a comprehensive, independent review of the available science. It rested almost entirely on two studies, both funded by the chemical industry, both designed to avoid the low-dose fetal exposure window, both reaching the expected negative result. The announcement was made in a sterile government conference room in Washington, D.C. The polished wood of the dais reflected the television lights. Agency officials spoke in the measured, reassuring cadences of regulatory science, the weight of evidence, the lack of a clear mechanism, the reassurance to consumers. A representative from the American Chemistry Council stood afterward and praised the FDA's thorough and science-based review. In the hallway outside, a small cluster of independent researchers, among them Vom Saal and a group of public health advocates, held a press conference of their own. Holding binders thick with studies the FDA had discounted or ignored. The television cameras briefly panned over them. Then the story moved on. What was not reported that day was that the FDA's own science board, an independent advisory body, had not been adequately consulted. When the board finally met several months later, it would deliver a verdict that shattered the agency's carefully constructed narrative. The assessment's flaws were not invisible to the scientific community. The National Toxicology Program, part of the National Institutes of Health, had been conducting its own multi-year review of the BPA literature. Its final report, released contemporaneously, expressed some concern about the chemical's effects on the brain, behavior, and prostate gland in fetuses, infants, and children. The first time a major federal health agency had used language that suggested caution. The discrepancy between the two government assessments was impossible to ignore. Then, in October 2008, the FDA's own science board met to review the draft. The meeting was held in a nondescript hotel ballroom outside Washington, the kind of space with chandeliers that had seen too many corporate retreats. It lasted for hours. One by one, the scientists dismantled the agency's methodology. They criticized the exclusion of dozens of peer-reviewed studies. They noted that the FDA had misinterpreted the industry-funded data, failed to apply appropriate statistical analyses, and ignored the core endocrine principle of low-dose sensitivity. The board's final report, released later that year, concluded that the FDA's draft assessment was inadequate, based on a limited and flawed set of data, and that the agency could not support its conclusion that BPA was safe. The polished wood of the regulatory fortress had developed a crack. The public, too, was beginning to move faster than the regulators. In 2007 and 2008, A wave of consumer pressure and investigative reporting pushed major retailers, Walmart, Toys R Us, CVS, to stop selling baby bottles containing BPA. Nalgene, the iconic outdoor gear brand whose translucent polycarbonate bottles were a staple of hikers and students, abandoned the material entirely. The same year, the Centers for Disease Control reported that BPA was detectable in the urine of nearly every American tested, a chemical fingerprint left by cans, bottles, and the slick paper of cash register receipts. The industry's carefully maintained front of scientific certainty was crumbling under parental anxiety, shareholder calls, and the simple optics of a product being pulled from shelves. Behind the scenes, something else shifted. Congressional investigators subpoenaed internal documents from the Weinberg Group and the American Chemistry Council, revealing the full architecture of the Doubt campaign. The records showed how industry consultants had ghostwritten scientific articles and paid for scientists to be present at regulatory meetings. They showed how trade associations coordinated press responses to bury damaging studies under preemptive rebuttals. The documents did not reveal illegal activity. Much of the conduct was technically within the rules of lobbying, but they exposed the apparatus for what it was: a calculated investment in uncertainty, funded at a level no academic researcher could match. The institutions designed to protect public health had used the authority of government to legitimize industry's version of the evidence, creating a perfect loop of manufactured uncertainty. The FDA's seal was affixed to science that had been bought and paid for, and the public was told the matter was settled. Frederick von Saul kept his laboratory running. The attacks on him subsided by the early 2010s as the public narrative evolved and the industry shifted to defending its replacement products. In his office, he maintained a file marked simply BPA History. It contained the original 2003 Weinberg memo, annotated with notes in blue ink. It contained the letters, the newspaper clippings, the press releases. The file was not a trophy. It was an artifact of a specific period in the history of American science, a period in which the very mechanisms designed to protect public health had been used to protect a product. The draft assessment of 2008 was never finalized. It remained suspended in an administrative gray zone, technically still the official draft, effectively abandoned as policy evolved. In 2010, the FDA updated its position, stating that it had some concern and supporting industry actions to stop producing BPA-based baby bottles. The language was circumspect. It did not admit error. It did not acknowledge the nearly 20 years of independent science that had been brushed aside. But it was a retreat. For nearly a decade, the United States government had told the public that BPA was safe. Then, an internal review quietly signaled a shift. The question of what finally moved the FDA does not have a single answer. It was not one study, not one hearing, not one courageous whistleblower. It was the slow, grinding pressure of accumulated evidence, the revolt of major retailers, the public's dawning unease, and the quiet but persistent voice of scientists who refused to let their data be buried. And yet, the story did not end there. Even as the FDA's language shifted, a new generation of chemicals was already rolling off production lines. The replacements bore names like bisphenol S and bisphenol F, structural analogs marketed under the reassuring label BPA-Free. The Weinberg Group had moved on to other clients. The American Chemistry Council had rebranded its website. The apparatus that had manufactured doubt about BPA was now, with the quiet efficiency of a photocopier, producing a fresh set of talking points for the next chemical. In the laboratory freezers at the University of Missouri, the tissues of von Saul's mice, from experiments conducted years earlier, remained preserved in their cassettes, the cellular architecture still readable under a microscope. The evidence was not going anywhere. Neither were the questions it posed. The last can of soup, the last thermal receipt, the last glossy BPA-Free sticker on a sports bottle, they all carried a single, unresolved pressure forward. The system had heard the warning, but it had not yet decided whether to heed it. In the spring of 2008, Anna Soto received an email from a former student that contained a link to a news article. The headline announced that Walmart, the largest retailer in the United States, would stop selling baby bottles and sippy cups made with bisphenol A. Soto read it at her desk at Tufts University School of Medicine, a room where for 17 years, every vessel, every flask, every pipette had been borosilicate glass. She had removed polycarbonate from her research in 1991, after the plastic leached a synthetic estrogen into her cell cultures and taught her a lesson she never forgot. The vessel was speaking the body's language, and no sterile protocol could silence it. Now the world was catching up. The recall did not begin in that laboratory, however. It began in a nursery, on a kitchen counter, in the steam of a bottle warmer. In 2007, an environmental health group called the Environmental Working Group published a report showing that BPA migrated from polycarbonate baby bottles into the liquid they contained, especially when heated. The report did not introduce new science. The science had been accumulating in journals for over a decade. What it introduced was a direct line from the molecule to the most vulnerable consumer, an infant drinking warm formula from a clear plastic bottle that had been advertised as sterile, modern, unbreakable. The images on television were not of cell cultures or rodent prostates. They were of ordinary kitchens where a parent poured hot water over a bottle to warm it, and in doing so, accelerated the leaching of a synthetic estrogen. The visual logic was swift and punishing. Within months, the polycarbonate baby bottle market, a symbol of mid-century convenience, collapsed. Toys R Us followed Walmart, then CVS, then a cascade of smaller chains. Nalgene, whose colored water bottles hung from backpacks across college campuses, pulled its entire polycarbonate line in April 2008. The chemical that had been invisible for 5 decades suddenly had a name the public recognized, and that name was enough to empty shelves. What was most remarkable was that no federal agency ordered the recall. The Food and Drug Administration maintained its 2008 position that BPA was safe at current exposure levels, a stance built significantly on industry-funded data. The recall was a market event driven by consumer fear and the retail calculation that a BPA-free label would sell more bottles than a silence. Safety was determined not by a regulatory body, but by a purchasing decision made in a grocery aisle. The market abhors a vacuum of convenience. The polycarbonate supply chain, worth billions of dollars globally, did not simply dissolve. It pivoted. Overnight, the term BPA-free became a marketing imperative. Manufacturers reformulated their plastics, substituting bisphenol A with a family of closely related chemicals that shared the same basic architecture, 2 phenol rings connected by a short chemical bridge. The most common replacement was bisphenol S, or BPS. Others included bisphenol F, bisphenol AP, and bisphenol AF. In the press releases and the glossy new packaging, these names were absent. The only name that mattered was the one that was missing. Bisphenol S was already produced in bulk for industrial uses, thermal paper coatings, epoxy resins, and as a monomer for certain plastics. It was cheap, available, and had a long track record in applications that never touched the human mouth. Its endocrine activity had simply never been investigated. The chemistry of the substitution was deceptively simple. Bisphenol A's central bridge is a propane group, two methyl groups that space the phenol rings at a distance that happens to fit the binding pocket of the estrogen receptor. Bisphenol S replaces that propane bridge with a sulfone group. The sulfur-oxygen bond is more polar, more heat-stable, and, in theory, less likely to leach under certain conditions. But the phenol rings remain. Their hydroxyl groups, which mimic the natural hormone's hydroxyl groups, still reach across the receptor cavity and form the hydrogen bonds that initiate a signal. The key is slightly different, but the lock does not require a perfect match. It only requires a persuasive imitation. By 2013, the paper that many scientists had been waiting for appeared in the journal Environmental Health Perspectives. Renee Vinyas and Cheryl Watson at the University of Texas Medical Branch reported that bisphenol S not only activated estrogen receptors, but did so with a potency comparable to BPA and through the same non-genomic pathways. Non-genomic signaling is the body's rapid response system. Estrogen docks at a receptor on the cell membrane, and within seconds, cascades of enzymes are mobilized. Traditional toxicology, with its focus on nuclear receptors and gene expression, had largely missed this dimension. Now, the replacement molecule was shown to trigger the same rapid cellular responses as the molecule it replaced. Subsequent research deepened the trouble. BPS resisted environmental degradation more stubbornly than BPA, which meant it persisted longer in rivers and sediments. It leached from replacement plastics at rates that could, under certain conditions of heat or acidity, exceed those of BPA. Some studies suggested BPS was more effective at disrupting thyroid hormone signaling or at interfering with the development of zebrafish embryos than the original compound. The replacement was not a correction. It was a lateral move into less charted territory. The marketplace was already filling with new products. Nalgene, the company whose recall had helped define the panic, switched to a new plastic called Triton, developed by Eastman Chemical. Triton was marketed as free of estrogenic activity, and its promotional materials cited proprietary testing. But independent testing by Certichem, a laboratory in Texas, reported that Triton products exhibited estrogenic activity in cell-based assays under certain conditions. Eastman Chemical sued Certichem, and although the lawsuit was eventually settled, the scientific dispute remained unresolved. The bright green sticker on a cloudy Triton bottle announced safety in the language of marketing: short, declarative, absolute. The science was less willing to use that full stop. In online forums and parenting groups, consumers traded the names of supposedly safe brands like evidence in a trial where the judge never arrived. The search phrase, "Is BPS safe?" typed into a glowing phone screen late at night, is the sound of a public trying to become its own regulator. The biomonitoring data from the U.S. Centers for Disease Control and Prevention bore out the biological uptake. By the mid-2010s, the CDC's National Health and Nutrition Examination Survey was regularly detecting BPS in the urine of roughly 80% of Americans tested. Simultaneously, BPA levels in the population began a slow decline, the result of consumer avoidance and manufacturer reformulations. The lines on the graph looked like a chemical swap, one bisphenol falling, another one rising. The body had not stopped absorbing a synthetic estrogen mimic. It had simply absorbed a newer one. Here was the paradox, as precise and profitable as any in industrial chemistry. In a desperate attempt to escape one endocrine disruptor, the market created mass exposure to a structural analog that had never been properly tested, repeating the exact same mistake with a different name. The replacement was already inside our bodies. The question returned, more urgent than ever. Are we doomed to an endless cycle of regrettable substitutions? In the years that followed, Ana Soto and Carlos Sonnenschein addressed this cycle directly. They wrote that the BPA-Free label was a semantic trick, a way of reassuring consumers while the chemical industry continued to produce close structural cousins with almost no independent safety testing. The regulatory system, they argued, treated chemicals as innocent until proven guilty. But the proof of guilt for endocrine disruptors required tools and concepts, low-dose effects, non-monotonic curves, transgenerational epigenetics, that the standard safety tests had never been designed to see. The system was blind by design, and the market was filling that blindness with new products. The physical record of this moment is not stored in a government vault. It sits on the shelf of a drugstore, in the baby aisle of a supermarket, inside the cabinet under a kitchen sink. The bright colors of sippy cups and water bottles are the dust jackets of an unfinished history. The receipts printed on thermal paper, still coated with free bisphenol molecules that slip into the bloodstream through the skin, fill wallets and glove compartments. The promise of BPA-Free was not a lie. It was a narrower truth than anyone wanted to believe. The industry had not been forced to prove safety before selling. It had only been forced to prove that one word was missing from the label. The other words, the other molecules, were still there, waiting for the body to read them. The experiment had not ended. It had simply been relabeled. The room smelled of cold ethanol and the faintly metallic tone that industrial freezers give off after years of continuous operation. At the NIEHS campus in Research Triangle Park, North Carolina, a row of freezers held the largest systematically collected library of human chemical residue ever assembled. Urine, blood plasma, breast milk, cord blood, and amniotic fluid, all time-stamped, all geocoded, all drawn from mothers and infants across the United States. The samples were not flash-frozen curiosities. They were a running tab of the chemical century, and by 2010, more than 93% of the entries in the column for bisphenol A came back positive. The cohort had begun in the early 2000s as an attempt to answer a quiet but persistent question: what are the levels of industrial chemicals in the bodies of ordinary Americans, and how do those levels change over time? The answer, for BPA, was both unremarkable and devastating. Unremarkable because the compound appeared in virtually everyone, at concentrations that toxicology textbooks considered biologically irrelevant, nanograms per milliliter, parts per billion. Devastating because, as Frederick von Saul had been demonstrating for over a decade in his laboratory at the University of Missouri, those levels were anything but silent. The strange thing was not that the chemical was present. The strange thing was that the toxicology textbooks had been measuring the wrong thing. They looked for gross damage at high doses, organ failure, atrophy, cancer, because that was the tradition of Paracelsus, the rule of the poison and its increasing shadow. But a hormone does not work like a hammer. It works like a question, and the body answers with an entire paragraph of physiological responses. A few molecules of estradiol, landing on a receptor in a fetal cell, could set off a chain of gene expression that determined whether that cell became a prostate duct, a mammary gland bud, or a neuron in a brain region that controlled appetite and anxiety. BPA, a synthetic estrogen rejected in 1936 for being too weak, turned out to be perfectly strong enough to ask the question in the wrong tissue at the wrong time. Frederick von Saal had first seen the generational echo in his own mouse colony in the late 1990s. He exposed pregnant mice to BPA at doses equivalent to those the CDC was now finding in amniotic fluid. The male offspring grew with enlarged prostates, and the female offspring matured earlier. That had been controversial enough. Industry-funded scientists called it impossible, a statistical artifact, a product of a contaminated lab. But von Saal continued the program. He let the exposed mice breed, then let their offspring breed, strictly controlling the diet and the cages to eliminate any other source of the chemical. What he found in the third generation, the grandchildren of the exposed dams, broke the simple arithmetic of toxicology completely. The grand offspring showed the same prostate enlargements, the same altered mammary gland structures, the same shifts in metabolic rate and sexual behavior as their grandmothers. Yet these animals had never been directly exposed. Their only contact with BPA had been through the germ cells of the embryo, a cellular whisper passed down the generations like a bookmark left in a gene. The route of inheritance was not a mutation in the genetic code. The sequence of DNA letters, adenine, cytosine, guanine, thymine, remained unchanged. Instead, the chemical had altered the way the genes were packaged and read. In the nucleus of every cell, the long strands of DNA are wrapped around proteins called histones, and small chemical tags, methyl groups, acetyl groups, are attached to the DNA itself, acting like quarantine flags or welcome mats for the cellular machinery that transcribes genes into proteins. When a methyl group locks onto a stretch of DNA near a gene, it can silence that gene, sometimes for the life of the organism. The process is called DNA methylation, and it is one of the central tools the body uses to specialize cells. A liver cell silences the genes for tooth enamel. A neuron silences the genes for stomach acid. BPA interfered with the placement of those tags, stripping some silencers away from genes that should stay quiet in the developing prostate, adding them to genes needed for normal brain development. In a famous mouse model, the agouti strain, a mother's diet rich in methyl-donating nutrients can turn her offspring's coat from yellow to brown by shifting methylation at a single gene. When researchers added BPA to the maternal diet, the offspring reverted to yellow, an almost shockingly visible proof that a synthetic chemical could override the epigenetic machinery. The effect was subtle, not a catastrophic malformation, but a shift in the boundaries of tissue growth, a widening of a duct here, an earlier surge of a hormone there. And the most disturbing finding, replicated in multiple laboratories by 2012, was that once a methylation pattern was imprinted by BPA in the germline, the cells that would become sperm or eggs, it could be inherited. The grandmother's brief encounter with a plastic can liner became a permanent annotation in the grandchild's physiology. The technology to read these tags had only become widely available in the late 2000s. Before that, the epigenetic markings were invisible, a molecular braille that no assay could translate. Now, a single chip could map the methylation status of 500,000 sites across the genome. The data poured out as heat maps, red and green blocks showing regions where the tags clustered densely, like storm systems on a weather map. In von Saal's mice, the storms consistently gathered around genes involved in cell proliferation and hormone signaling. The first clear demonstration that an endocrine disruptor could rewrite heritable methylation patterns had come 5 years earlier from a laboratory at Washington State University. Michael Skinner exposed pregnant rats to the fungicide vinclozolin in 2005 and found prostate disease and sperm deficits not just in the offspring, but in the great-grand offspring, accompanied by a stable set of methylation changes. The paper, published in Science, shattered the assumption that environmental effects washed out with each generation. Vom Saal saw the Skinner data and recognized the pattern. His own mice had been telling the same story, but without the molecular signature. By 2008, his group and others had confirmed that BPA did the same, at doses the human biomonitoring data suggested were already inside us. On the screen in Vom Saal's office, a generated model of the human estrogen receptor rotated slowly, a ribbon-and-pocket structure of glowing amber loops. The receptor is a protein shaped like a cupped hand, waiting for the arrival of estradiol, the natural ligand. BPA, the molecule that started life as a condensation product of phenol and acetone, was smaller and flatter than estradiol, but it could still slip into the pocket. The fit was imperfect, but in the low concentrations of the fetal environment, the receptor had no better occupant. It bound, and the signal cascaded outward. Genes switched on, proteins assembled, cells divided. The receptor could not distinguish between the body's own hormone and the molecule drifting up from a polymer. That molecular promiscuity had been controversial for a decade. The chemical industry pointed out that BPA's affinity for the receptor was thousands of times weaker than estradiol's. But Vom Saal and others had shown that the fetal proteome, the entire set of proteins circulating in development, was effectively a quiet room. There was so little competing estradiol that even a weak signal became the dominant one. In the womb, BPA wasn't a whisper. It was the loudest voice in the room. The proof that this mechanism operated in humans would never come from a controlled experiment. You cannot deliberately dose pregnant women with an endocrine disruptor. But the evidence accumulated from epidemiological studies that used the NIHS biobank samples, correlating BPA levels in urine and cord blood with health outcomes years later. By 2016, a comprehensive review of those studies had linked fetal BPA exposure to altered neurodevelopment, language delays in girls, hyperactivity in boys, to increased body fat mass, to markers of metabolic disorder in childhood. The statistics were not unanimous, but the weight of the animal mechanism and the human biomonitoring data made a coherent argument. The lowest dose, delivered during the narrow windows of organ formation, was the one that mattered most. The fetal origins of adult disease, a hypothesis first proposed by the British epidemiologist David Barker, had grown from a niche theory into a core principle of 21st century medicine. Barker had shown that babies born underweight had higher rates of heart disease in middle age, as if the fetal body, deprived in the womb, had made metabolic trade-offs that became bad bargains later. BPA extended this principle into the realm of endocrine mimicry, the fetal body. Mistaking the plastic-derived signal for a real hormonal instruction, made permanent adjustments to its receptor sensitivity, its fat cell number, its brain circuitry, adjustments that would only show their cost decades later. The NIH's freezers stored not just vials, but evidence of a vast unlegislated experiment. Each barcode on each tube represented a person who had never consented to be a subject, a mother who had shopped carefully, a family who had used BPA-free bottles, but whose century-old food supply still arrived in epoxy-lined tins, whose receipts still coated their fingers with free unbound monomer. vials sat in numbered racks, the codes on their labels replacing names, connecting to medical records in a secure database, but in the cold light of the freezer aisle meaning nothing, just another aliquot of the chemical century. By 2020, the cohort had spawned more than 200 published studies, each one peeling back a layer of the fetal exposome. The biomonitoring data showed that BPA levels in the population were declining slowly after the 2009 bans on baby bottles, but they were being replaced by bisphenol S and bisphenol F, chemicals with similar structures and, the early data suggested, similar epigenetic capacities. The archive did not close, it updated. In the animal models, the grandmother's exposure became the grandchild's physiological debt. The question no epidemiological study could yet fully answer was, what equivalent debt were the human generations carrying? The first epidemiological cohorts of children born in the 2000s were just reaching adulthood. Their metabolic profiles, their fertility, their neurological baselines were only beginning to be written. The experiment was still running, and the results, for another 20 years at least, would remain inside the bodies of millions who had never heard of a bisphenol. The evidence is not buried in a landfill or dissolved in a riverbed. It circulates in the bloodstream of every industrialized population on Earth, filed under a barcode in a cold corridor in North Carolina, and in the methylome of the next generation's germ cells. We are the archive. The pages are still being printed, and the ink of the plastic century will take 2 more human lifetimes to fully dry. And yet the archive remains stubbornly illegible in parts. For every epidemiological study that links prenatal BPA to altered neurodevelopment, another, equally rigorous, finds no significant association after adjusting for household income and maternal education. The signals are small, the confounders are many. A pregnant woman is not a laboratory mouse. She encounters a chemical mixture, phthalates, flame retardants, pesticides, that may amplify or cancel BPA's epigenetic mischief. The archive holds the truth, but its glyphs are written in overlapping inks, and the key to reading them will take still more time to cut. The dimpled metal of a can of pumpkin puree holds the fluorescent light of the grocery store like a mute confession. The cans stand in orderly rows, their labels bright with autumn illustrations, a farmhouse, a wedge of pie, promising a harvest that will taste the same in February as it did in October. The metal is cool, the seams precise. It is an object engineered for trust. And yet, bonded to the inner surface, invisible and unacknowledged, is a skin of amber resin thinner than a human hair. That skin carries a molecule that, in a different vessel, once impersonated a hormone in a petri dish in Soto's laboratory. The year was 1991. Soto and her colleague Carlos Sonnenschein at Tufts University School of Medicine had been growing breast cancer cells in a medium designed to be estrogen-free. The control cultures, meant to languish without growth signals, began to multiply. The contaminant was not an added chemical, but a substance leaching from the polycarbonate flasks themselves. The flask was supposed to be sterile, inert, a neutral container. It was not. It was delivering a microdose of bisphenol A into the culture medium, a molecule whose shape allowed it to slip into the estrogen receptor and flick the switch. The discovery was a laboratory accident that would ripple outward over the next quarter century to touch every can of soup, every receipt, every infant's bloodstream. By the end of the first decade of the 21st century, that molecule had been banished from the nursery. In 2008, after a series of damning studies and a consumer panic fueled by the phrase, gender-bending chemical, retailers began pulling polycarbonate baby bottles from their shelves. In 2009, the 6 largest manufacturers of baby bottles in the United States voluntarily agreed to stop using BPA. In 2012, the FDA amended its food additive regulations to formally prohibit the chemical in baby bottles and sippy cups. It was a landmark moment. The concerned parent could now purchase a bottle stamped with a green leaf and the words, BPA-Free. The shelves of the infant aisle had been cleansed. But the can did not follow. The canning industry had been applying BPA-based epoxy liners to the insides of steel and aluminum containers since the 1950s. The technology had solved an ancient problem. Before epoxy, canned goods were a gamble. Acidic foods, tomatoes, citrus, sauerkraut, corroded the tin coating that protected the steel. The resulting pinprick holes could admit bacteria, including Clostridium botulinum, whose toxin paralyzes muscles and stops breathing. The epoxy liner, developed independently by chemists at Shell and Ciba, formed a continuous, virtually impermeable barrier. It was cheap, fractions of a cent per can, and it survived the 250-degree retort that sterilized the contents. The FDA classified the resin as generally recognized as safe under a regulatory framework that asked a single blunt question: does it cause acute poisoning? It did not. The question of whether it might, at vanishingly low concentrations, speak the body's hormonal language was not on the exam. It was not even a phrase that the regulators of the Eisenhower era possessed. The technical challenge of replacing an epoxy can liner remains immense. A can's interior is a corrosive chamber. Tomato sauce, with a pH near 4, eats at steel like a slow acid bath. Carbonated beverages, even more aggressive, hunt for microscopic breaches. The liner must adhere to the metal without flaking, flex with the can's expansion during heating, and maintain a flawless seal for up to 5 years on a grocery shelf or in a pantry. It must not impart taste. It must not dissolve into the food. BPA-derived epoxies meet these demands with the precision that borders on molecular architecture. The polymer forms a dense, cross-linked network that resists acids, alkalis, and mechanical stress. But no polymerization reaction is perfect. A residual fraction of the starting material, the bisphenol A monomer, remains unreacted. It lingers in the resin matrix as a free molecule, available to migrate into the food the liner was designed to protect. And migrate it does. The rate of leaching increases with heat and acidity. The exact conditions inside a can of tomato soup, freshly pressure cooked and sealed, are ideal for liberating the monomer from its cage. On the floor of a canning plant in Modesto, California, the line hums a deep industrial note. Empty cans clatter down a conveyor, are filled with diced peaches in heavy syrup, capped, and shunted into a pressure cooker the size of a delivery truck. The superintendent has worked this line for 30 years. He knows the liner by its functional alias, the coating. He has never seen a can fail from corrosion since they switched to this coating in the 1960s. The alternatives he has read about, acrylics, polyesters, have a reputation for stress cracks, off flavors, customer complaints. He trusts the coating because it works. The invisible residue that might ride the peaches into a child's lunchbox is not part of his daily calculus. It is a problem that happens in parts per billion, in university labs, in regulatory hearings a continent away. In the spring of 2015, the Natural Resources Defense Council petitioned the FDA to revoke all regulations permitting BPA in food packaging. The submission cited the now vast scientific literature descended from Soto's accident, thousands of studies mapping the chemical's affinity for estrogen receptors, its effects on mammary gland development in rodents, its links to metabolic disruption, altered brain architecture, earlier puberty in females, reduced sperm quality in males. The Endocrine Society, the American Medical Association, and the World Health Organization had all expressed concern. The European Union had banned BPA from baby bottles in 2011. France had extended the ban to all food containers. Anna Soto, now a recognized authority on endocrine disruption, joined her colleagues in urging the FDA to adopt a precautionary framework to treat the molecule's mimicry as a public health threat. The FDA, on December 31, 2015, denied the NRDC petition. The agency's response was a careful, legalistic document. It acknowledged that BPA is present in the urine of nearly all people tested. It agreed that some studies have reported adverse effects, but it concluded that the evidence does not provide sufficient basis to withdraw the approval. The framework the FDA uses for food contact safety is a product of the mid-20th century. It is built on Paracelsian toxicology. The dose makes the poison. It requires a clear, monotonic relationship between exposure and effect. It cannot easily accommodate the non-monotonic endocrine curve, where low doses cause effects that higher doses do not, that Frederick von Saul, Soto, and others had documented. The agency, faced with a science that did not fit its tools, chose to trust its tools. In a kitchen in Ohio, a woman opens a can of tomato soup. The can opener's rotary blade cuts the seal with a soft click and a hiss of vacuum. She pours the concentrate into a saucepan, fills the empty can with milk to rinse the last redness from the liner, and stirs. The soup is for her 7-year-old daughter who is drawing at the table. The mother has seen the BPA-free stickers on plastic water bottles, but she does not think about the can in her hand because cans are metal. The label does not mention epoxy. The exposure, if it occurs, is invisible, undetectable to the tongue, and probably, she tells herself, too small to matter. The soup is warm and familiar. Her daughter dunks a triangle of grilled cheese and laughs at the orange smear it leaves. The regulatory stalemate opened a lateral battlefield, the courts. Starting around 2010, plaintiffs' attorneys filed class action lawsuits against food companies and can manufacturers, alleging that the presence of BPA in food packaging was a material omission under state consumer protection laws. In 2013, a suit against ConAgra Foods over its canned tomatoes settled with an agreement to explore alternatives. Campbell's Soup Company, facing its own litigation, and a coalition of shareholders, announced in 2016 that it would transition away from BPA in all its cans by a future date, a process that took years and required reformulating liners, testing for taste and safety, and retrofitting factories across North America. Smaller, nimble brands moved faster. Eden Foods had switched to amber glass jars for its beans as early as 1999. A handful of startups began packaging soups and broths in aseptic cartons, the kind used for juice boxes. Grocery chains like Trader Joe's and Whole Foods pressured suppliers to adopt BPA-free cans. The market, in its fragmented, contradictory way, began to inch past the deadlocked bureaucracy. But the replacement liners carried their own quiet liabilities. The most common substitutes were acrylic epoxies, polyester films, and oleoresins. None performed as reliably as the BPA-based epoxy. Acrylics were prone to stress cracking during the heat treatment process. Polyester could delaminate in sheets. Oleoresin, a natural resin derived from tree sap, could not withstand the acidity of tomatoes or the carbonation of soda. And the raw materials for the synthetic replacements, the acrylic monomers, the polyester precursors, the cross-linking agents, had undergone even less scrutiny for endocrine activity than the bisphenol they replaced. When researchers tested some BPA-free can liners, they found bisphenol S or bisphenol F, structural cousins with similar estrogenic potency and, in some cases, even greater leaching rates. The cycle of regrettable substitution, which had already galloped through the plastic aisle, was now playing out inside the can. The can is the final frontier because it crystallizes a systemic deadlock. Industry wants a drop-in replacement that is cost-neutral, performs identically, and carries no PR risk. Regulators want unequivocal proof of harm that fits a 20th century test matrix. Scientists want the precautionary principle, but they speak in probabilities and biological plausibility, not certainties. Consumers want to feed their families without fear, but they do not want to read a PhD dissertation every time they shop. And so the cans continue to roll off the lines, an estimated 300 billion of them annually worldwide, their interiors glazed with a molecule that a Russian chemist first synthesized in a flask in 1891, that a British biochemist briefly considered as a medicine in 1936, and that industrial chemistry resurrected as a miracle of mid-century materials. In Boston, Anna Soto keeps the original polycarbonate flask on a shelf in her office, a souvenir from the experiment that changed the trajectory of her career, and, slowly, the regulatory posture of nations. Her laboratory now uses only glass and stainless steel. The borosilicate is heavy, impermeable, silent. It does not mimic. It does not leach. It is a deliberate retrenchment, a return to materials that do not speak the body's language. But outside that room, the cans are still being filled. The liners hold. The soup is heated. The body receives its daily, unlabeled dose. The new liners are already in circulation, their toxicology files thinner than the films they deposit on the metal. The next generation of biomonitoring data will read what they have written in the blood and urine of millions. For now, the can opener turns, the metal teeth bite the rim, and the experiment continues, without a control population, without a clear hypothesis, and without the informed consent of those who are, every day, the subjects. The flask is a simple thing, borosilicate glass, heavy in the palm, its walls cool and impermeable. In the laboratory at Tufts University School of Medicine, Anna Soto sets it down on a steel bench. The glass does not speak. It does not leach. It does not whisper to the cells inside. There was a time 30 years ago when every vessel in this room was polycarbonate, clear, shatterproof, a triumph of 20th century chemistry. That was the problem. The E-screen assay Soto and her colleague Carlos Sonnenschein had built was meant to detect estrogenic activity. The control cultures were supposed to be silent. They were not. The cells multiplied as though dosed with hormone, and the source was not a chemical added to the medium. It was the flask. The container itself was speaking the body's oldest language. Today, every flask, every beaker, every tube that touches a cell in Soto's laboratory is glass or rigorously tested polypropylene. The change is absolute. It is also, in its own way, a quiet indictment of a regulatory system that still permits the old materials to touch food, to line tin cans, to coat thermal receipts. The laboratory purified itself because the science demanded it. The wider world, for reasons less straightforward, has not. The years between that discovery and this glass room have been dense with argument. The American Chemistry Council orchestrated a defense of BPA that ran on 2 tracks: fun studies designed to find no effect, and frame the scientists who found effects as activists. Stephen Henches, the industry's voice, sat in polished hearing rooms and delivered testimony that parsed uncertainty into a shield. The FDA, in 2008, declared BPA safe, leaning heavily on industry-funded data and rejecting the low-dose hypothesis that Soto's work had helped birth. Frederick von Saal, whose mouse studies showed changes at parts per billion, became a target of personal attack. The institutions that were supposed to guard the public instead guarded the status quo. The machinery of doubt worked as it was designed to work. It bought time. It kept the canning lines running. It held off the question that the E-screen had posed in 1991. What does it mean when a material that wraps our food can also dock with a nuclear receptor and change the expression of a gene? In 2016, something shifted in the architecture of American chemical law. The Frank R. Lautenberg Chemical Safety for the 21st Century Act reformed the Toxic Substances Control Act, a statute that had been a monument to paralysis since 1976. TSCA's original language grandfathered in 62,000 chemicals without requiring safety data. The Lautenberg Act gave the EPA new authority. It had to evaluate existing chemicals, prioritize the most dangerous, and, crucially, consider vulnerable populations like pregnant women and infants. It required the agency to weigh endocrine disruption. It was a step, a genuine step, but a step is not a landing. The law did not ban BPA from food packaging. The FDA would not do that for another 7 years, and even then, the change would come not through legislation but through consumer pressure and corporate pledges. The can of soup on the grocery shelf still carries a lining derived from a bisphenol, though the label no longer names it. The receipt handed to you at the register still carries its powdery coat of free monomer. The replacements are structural analogs. BPS, BPF, siblings of BPA, built from the same functional family, carrying the same 2 phenol rings, docking at the same receptor with only slightly altered affinity. The cycle of regrettable substitution grinds on. The public learned that the materials protecting our food could speak the language of our hormones. The panic of 2008 was real. The baby bottle recalls were real. The eruption of BPA-free marketing was real, but the reckoning was never completed. The system that produced the problem, a chemical industry that invents faster than it tests, a regulatory apparatus that demands proof of harm while permitting exposure, a consumer culture that wants convenience without consequence, remained largely intact. We swapped molecules, but left the machinery running. In a kitchen, someone fills a glass with water from a pitcher. The pitcher is plastic, stamped with the green leaf of a BPA-free claim. The pipes running from the municipal water system are PVC or HDPE. The water carries a faint fingerprint of the molecules this century has chosen to mass produce, not just bisphenols but phthalates, parabens, the long tail of industrial chemistry that has entered the hydrological cycle. The drinker swallows. The liver conjugates what it recognizes as foreign. The remainder circulates. The National Health and Nutrition Examination Survey has measured it for decades. BPA in over 90% of Americans. BPS now rising in the same samples. The body is a rolling archive. And the archive is not merely chemical. It is epigenetic. Studies that began in Soto's early collaborators' laboratories and that now span institutions across 3 continents have shown that endocrine disruption can alter the methyl marks on DNA, the chemical annotations that tell a cell which genes to read and which to silence. The marks survive cell division. In animal models, they survive generations. A rat exposed in the womb can pass a changed chromatin landscape to its grandpups. The exposure does not need to be large. The dose that matters is the dose received during the brief, plastic window of development when the body's wiring diagram is being drawn. The chemical industry fought for decades to prove that BPA levels were too low to matter, that the parts per billion concentrations in human urine were trivial. The biology now says otherwise. The timing is the dose. Soto keeps the early papers in a filing cabinet in the department archive. A postdoctoral fellow opens the drawer. The 1991 manuscript is flagged with yellowed tape. The tables record cell counts from culture wells that should have been controls. The numbers rise unexpectedly, consistently, across multiple experiments. The handwriting in the margins is Soto's. It records the moment when a century of polymer chemistry collided with a century of endocrinology, a collision that no one had planned, that no safety test had anticipated, that hid in plain sight inside a sterile incubator. The paper smells of aged cellulose. The ink is stable. The truth it contains has not changed. The sound of a tap running water through plastic pipes into a glass basin is the sound of the modern world. It is a sound so ordinary that it rarely registers. In Soto's laboratory, the water passes through a filtration system and emerges from a stainless steel faucet. The glassware is washed in glass beakers, dried in a glass-walled oven. The protocols are strict. The goal is to banish the background noise of plastic so that the cells can tell their story without a second script already running. The irony is palpable. To study how the body responds to its chemical environment, you must first remove the chemical environment that modernity has made universal. The burden of proof remains a battleground. In the United States, the default assumption is that a chemical is innocent until proven guilty, and the proof, by design, is immensely difficult to assemble. Epidemiology moves slowly. Endpoints like cancer or infertility take decades to manifest. The tools for measuring epigenetic change are still sharpening. The precautionary principle, the idea that potential harm should be prevented before full scientific certainty is achieved, has guided European chemical policy since the REACH regulation of 2006. In the US, it appears more often in dissenting opinions than in binding statutes. The Lautenberg Act was a departure, but not a conversion. It did not flip the burden. It did not demand that new chemicals be proven safe before they enter the market. It simply modernized the assessment process. The ghost of BPA remains the template for the next 1,000 substances that will arrive in the coming decades, each with its own quiet affinity for a receptor no one thought to screen. Anna Soto's glass room sits inside a larger building, which sits inside a city, which sits inside a regulatory ecosystem that has not yet decided whether the convenience of plastic is worth the cost. The laboratory is a choice, a deliberate act of refusal. Refusal to trust the composite. Refusal to accept that the materials of industry must also be the materials of biology. But the choice is not widely available. Most of the world cannot run its experiments in glass. Most of the world must eat from cans, drink from bottles, touch receipts, and trust that someone, somewhere, has done the accounting. The accounting, however, is incomplete. The ledger of endocrine disruption is still being written. The biobank freezers at the National Institute of Environmental Health Sciences hold rows of labeled vials, blood, urine, breast milk. that track the chemical body burden of the American population across generations. The data from those freezers show that the plastic decade, the 1950s onward, left a residue that does not fade when the product is thrown away. The polymers degrade, but the monomers persist. They cycle through water, dust, food, air. They cross the placenta. They enter the breast. The first meal a newborn receives, if it is breastfed, contains a dose of the industrial 20th century. The second meal, if it comes from a polycarbonate bottle, contains another. The question that remains is not whether BPA was a mistake. The question is much larger. It is whether a system of production that invents a new chemical every 9 seconds, an estimate from the American Chemical Society's own registry, which now catalogs over 200 million substances, can ever be matched by a system of testing that takes years to clear a single compound. The gap between invention and understanding is not a crack. It is a canyon. And across that canyon, a line of molecules waits its turn, each one hoping to become the next invisible backbone of daily life. Soto lifts the glass flask. The light passes through it cleanly. There is no cloud, no residue, no latent signal waiting to emerge under the right conditions. The polymer that once filled this room with its hormonal mimicry has been banished. But the flask is also a symbol, and symbols can be deceptive. They suggest that the problem has been solved because a single laboratory has insulated itself. The real problem never lived in this room. It lived in the patent offices, the chemical plants, the regulatory conference rooms, the supermarket aisles. It lived in the assumption that the chemistry of convenience could be separated from the chemistry of the body by a wall that would never crack. The wall did crack. The crack was found by a woman culturing cells in plastic, expecting silence, hearing speech. That crack has widened over 30 years into a public debate, a body of science, a partial reform, a marketing shift, and an unanswered question. The plastic that learned to imitate hormones was not a villain. It was a molecule that did exactly what its structure allowed it to do. The failure was not in the chemistry. The failure was in the systems of oversight that assumed a molecule could be judged by its immediate toxicity alone, that a dose-response curve was the only language of harm, that the womb was not a listening chamber. The story is not over. The archive is still open. The next chapter is being written right now, in the choices made in laboratories like this one, at cash registers where receipts are handled, in legislative sessions where chemical safety is debated, in homes where parents reach for a bottle marked free of one compound, unaware that its replacement has never been scrutinized. The glass room is a beginning, not an end. It is a lesson in humility, a demonstration that foresight is cheaper than repair, that silence from a flask is worth whatever it costs to achieve. The question is whether we are ready to learn that lesson before the next chemical arrives, before the next miracle of convenience is molded, shipped, wrapped around our food, and handed to us without a whisper of warning. The polycarbonate flask sits on Anna Soto's shelf in Boston, a souvenir from the moment a sterile vessel began to speak. Inside it, cells multiplied without permission, and the container that was supposed to be neutral had been whispering a hormonal command all along. That flask became the first piece of evidence in a long, slow investigation. The glass room of her laboratory, where every culture vessel is now borosilicate, stands as a deliberate refusal, a retreat from a century of materials that had never been tested for the language they carried. And in the freezers of the National Institute of Environmental Health Sciences, Row after row of labeled vials hold the residue of that century. Urine, cord blood, breast milk, all of them marked by the same molecule in 93% of those tested. The truth that became visible is that a substance can be useful and dangerous at the same time, and that the danger can operate at a dose so low it was never looked for. The regulatory system, built on the assumption that harm scales with quantity, could not detect the chemical that worked through a receptor already tuned to whispers. The cost of that blind spot was not a single accident, but a slow, universal exposure, passed from mother to child, from can to kitchen, from receipt to fingertip. The evidence changed the understanding of what safety means, not as the absence of acute poison, but as the silence of the container. Now the pressure eases from the hull. The darkness thins above, first to a deep blue, then to gray, then to the pale green of light breaking through. The hatch opens, and the cold sea air enters the lungs. The archive file closes, but the recovered evidence remains with us. Some substances are safest when they stay in the archive, not in the living body. The question that floats up with us is not whether we learned a lesson, but whether we will apply it to the next molecule before it is already inside us. If this kind of quiet descent into science and history is what you came for, you can subscribe to the Deep Archive. The surface returns, and the glass flask stays on the shelf, no longer an accident, now a document.